Good evening, everyone. Welcome to Byju's Exam Prep IS. Before we start, if the audio and video both are working fine, please let me know. Please give me a confirmation. All good? Wonderful. So, today we are going to discuss and start from where we had stopped last class, and we had started our chapter on biotechnology, where we had discussed the aspects of medical applications of biotechnology. Now, that is where we start from, and today we are looking at agricultural applications of biotechnology as our first chapter. So we understood that majority of the money that flows in biotechnology, it flows in the medical science, and that too specifically in uh, biopharmaceutics. So in biopharmaceutics, we see that majority of money flows. But apart from that, if there is a sector where a lot of research is being done and a lot of emphasis is being put, and biotechnology has been trying to make a change, is agriculture. And in agriculture, we have seen several instances where, in one way or the other, biotechnology has been very, very helpful. Now, let's try to understand all those aspects. And one of the most important, uh, and you can say one of the success stories of agriculture has been this. So, do you know what exactly this is? Any idea what this is? Cotton, yes. And specifically, which cotton do you know? Since we are talking about biotechnology, any idea? Right, this is about BT cotton. So, this basically is BT cotton. Now, when it comes to BT cotton, in case of BT cotton, what we understand is that this has been one of the ways in which biotechnology has done something immensely important for agriculture. Now, what we saw almost 25 years back in cotton was that cotton crops in India were suffering from a certain pest, and this pest was bollworm. Now, this bollworm would eat upon the flowers of cotton and they would completely destroy the cotton crops. And approximately 25 to 30% reduction would be observed in the productivity of cotton. So, now this was hampering the agriculture a lot, and productivity was low. Farmers were uh, seeing a lot of issues because of this. So this is where we saw that there was one change that was done, one modification that was done in the cotton plants. And this change that we did right here was introduction of a certain bacteria. A certain bacteria was added to the cotton plant, and this bacteria was Bacillus thuringiensis, called as BT. Now, this BT was a bacteria. And as we added bacteria to this cotton, the genes of bacteria basically, the genes of bacteria were added to the cotton. And as soon as genes of bacteria were added, we saw that cotton now would be able to make its own pesticide. All right. It will be able to make its own pesticide. And by making this pesticide, the cotton crops will become poisonous for bollworm. And this is where we saw that we were able to increase the productivity of cotton that had dropped by 25 to 30%. So this was, you can say, one of the success stories of biotechnology in agriculture. Now, when we talk about this kind of an arrangement where you are introducing a certain foreign gene, meaning a gene which is coming from some other organism, maybe a microorganism, maybe a plant, maybe an animal, wherever it is coming from, when you have a foreign gene in a certain plant, that kind of plant will be called as a transgenic plant, or crop will be transgenic crop. So that's where this term transgenic comes from. And that's why when you use the word trans, it will be used in a manner where something is coming, or there is something that has come from somewhere else. For example, even if somebody goes from one place to another, we say that the person has been transferred, right? We say that the person has been transferred. So that means what? That person now has moved from one place to another. So similarly, when we say transgenic, it means that there is the presence of a trans gene, meaning that there is a gene coming from another organism. And this can be any organism. Just remember this, Very, very important, that this can be any organism. There is no restriction that if you are talking about a plant, only a plant gene can be taken. No, even if it's a plant, you can take an animal gene, you can take a microorganism gene. For example, in bt cotton, what have we done? We have taken the gene from a bacteria. So you can take a bacterial gene, you can take a viral gene, you can take a gene from an animal, you can take a gene from a human. If you want, you take a gene from an alien, if you can. So you can take the gene from anywhere. The only meaning of transgenic or transgene is that this gene should be coming from some other organism, and we have no definition for what organism it should come from. Any organism is fine. All right. So that's why in transgenic crop plants, one or more genes are artificially inserted instead of the plant acquiring such genes through pollination. So this is where the genes will come from some other plant, and that's why it will, or some other organism for that sense, and that's why it will be called as a transgene. So transgene basically will be gene taken from some other organism. Right, gene taken from some other organism will be called as a transgene. Now overall, when it comes to this transgenic plant, these kind of crops will also be called as genetically modified crops. All right, genetically modified crops are sometimes also called as genetically modified organisms, or gmos. So gmo means genetically modified organisms. So now when it comes to these transgenic plants would have a lot of different types of desirable features. And these are desirable features like, for example, increasing the crop productivity. What we discussed in the case of BT cotton, that how we were able to increase the productivity of cotton by introducing this certain gene. So that's why BT cotton, we see this particular gene which has a better yield altogether. Then apart from that, we also see that it can help in improving the nutritive value. Now, how it can improve nutritive value, there also we have certain examples where we have seen that even doing this is possible for us, that we can improve the nutritive value of any given plant. So one prime example here is of golden rice. Now, this rice that we have here, this is called as golden rice, and this is the normal rice. Now understand what the difference between the two is. What's the difference between golden rice and normal rice? Now golden rice basically is the rice, as you can see that it is yellowish in color, but yellow rice would not sound good, so we named it as golden rice. So golden rice basically, as you can see that it has a different color. Now overall what we understand what we understand is that this golden rice basically is something where you have extra vitamin A. All right, we have extra vitamin A in golden rice. Now in case of golden rice, how this extra vitamin A comes is basically from another plant, that is the daffodil plant. All right, there is the daffodil plant, and we take something called as beta carotene from this daffodil plant. And this beta carotene, the genes of beta carotene, when inserted inside rice, this is where we see that this rice will now start having its own vitamin A. It will start producing its own vitamin A. So now what happens is that golden rice will have enhanced amount of vitamin A. All right, it will have enhanced amount of vitamin A. So otherwise, when you consume rice, you don't consume rice for vitamins, right? You don't consume rice for vitamins. You consume rice largely for carbohydrates, right? Rice will give you carbohydrates. So this carbohydrates is where we see that we would see generally we would consume rice for. But golden rice is specifically, golden rice specifically will have enhanced amount of vitamin A as well mixed in it. So this is where we see that golden rice becomes a transgenic variety of rice with genes for synthesis of vitamin for beta carotene taken from the daffodil plant. All right, and that's why beta carotene is converted into vitamin A. So this beta carotene is converted into vitamin A, and this is how we would see an improvement in altogether in the nutritive value. So that's why this is also possible. Similarly, we see that there are other applications, like we can increase the shelf life. We can also see environmental benefits here. So there are all these ways in which uh, it can help. So for example, let's say when we use bt cotton, so use of bt cotton also means that uh, you do not have to use chemical pesticides externally, correct? You do not have to use it externally. You can because the plant is itself able to produce its pesticide, correct? So in that sense, what happens is that there are environmental benefits, that these chemical pesticides which could have been very dangerous, they actually they actually will be able to save them. So that's why environmental benefits also, an improved shelf life, in the sense that when it comes to uh, many of the seeds or even fruits or vegetables, we can enhance their life cycle, we can enhance uh, their quality altogether, so that they are not getting affected by pests, they are not getting affected by germs or microorganisms easily, and they would last longer. And that's why we'll be able to improve their shelf life altogether. So that's why there are all these ways in which we can make the use of uh, transgenic plants. Now, when it comes to the development of transgenic plants, how do we do that? We are saying that, okay, fine, there is a gene, and we will insert that gene inside the body of this organism. Now, how exactly do we do that? Is that, I mean, is it even possible to do something like this? Can we do something like this? Or is it a very simple method that we just have to choose a gene, insert it inside the plant, and we are done? Unfortunately, things are not that simple. You cannot simply just choose a gene and just insert it in the plant and it starts working. It's the things are not that simple. Now, when it comes to the understanding of how things work, there is this process which is called as recombinant DNA technology. All right, this is called as recombinant DNA technology. Now, in case of recombinant DNA technology, understand what recombinant DNA technology says, how it works. So overall, the things are not that simple. As I told you, that you cannot simply just pick a gene and you can insert it and it starts working. Then in that case, we could have made a plant walk, that you just find the genes which humans have, which make us walk easily. You just find those genes, and once you have those genes, you simply have to insert it inside the plant. And once you insert it in the plant will start walking. But unfortunately, things do not work in that fashion. So why it doesn't work in that fashion? Because there is this entire process that we have to use to ensure that this recombinant DNA technology works. Now, how exactly do we make this work? So first, what we do is that we will take the gene of interest. We'll take the DNA that contains gene of interest, and once we have that, what is done is that this gene will be taken, and this gene will be inserted inside another carrier. We will choose a carrier, or also called as a vector. So we choose a carrier or a vector. All right, we choose a carrier or a vector. Now, this carrier or vector, this can be either, in most of the cases this will be a virus, most of the cases it will be a virus, but it can also be a bacteria. All right, it can also be bacteria or it can be virus. So either virus or bacteria will be chosen as a carrier. Carrier of what? Carrier of this gene, the gene of interest, the bt of bt cotton, or the beta carotene containing gene that we inserted in golden rice. So our gene of interest, first what we'll do is that we'll pick the carrier. Once we have picked the vector, this is where we'll do this, that we will take this gene and this gene will be inserted inside. Now, even this insertion is not that easy. You first have to find exact matching place where this gene can be inserted. It's just like a jigsaw puzzle, that you can only fit the piece of puzzle where it belongs. You cannot simply place it anywhere. All right, you cannot simply place it anywhere. You have to pick this place where it belongs, and you only have to insert it there, and you cannot insert it anywhere else. So something similar has to be done here, that in this vector also you have to see where exactly can you insert this. And once you have found that place, and there are genetic tools that we have, there are genetic engineering tools that we have, and with the help of these genetic engineering tools, we find where exactly can we do this, where exactly can we make this insertion, where exactly can we do this. All right. So for that, what we do is that we will find the place, and this will be done with certain enzymes. For example, there is something called as restriction enzymes. So these restriction enzymes will go and identify the place where the cut has to be made, and they will make the cut. So once they have made the cut, it will be something like this, you can say. Once they have made the cut in this, it will be like this, right? So we have made the cut here. As you can see, the cut is being made here like this. The cut is being made here. So once the cut is made, it will open into two different pieces. It opens into two different pieces like this. And once it has opened into these two pieces, this is where we insert our gene of interest. This is where we will insert our gene of interest inside this vector. Right, like this. So this is how you have the things, that this is the vector, this is the vector gene, and this is our gene of interest. So this is our gene of interest, and vector gene is different. So now what happens is, once you've inserted it, you now have to stick the ends together. So you need a favicol or a favistick, correct? So I'll go to the shop, I'll buy the favistick, but unfortunately favistick doesn't work. So now this thing that you are using here to bind these things together, it is called as DNA ligase. This is called as DNA ligase. So overall, we see that this is how now we create something which is called as recombinant DNA. Now this DNA that we have right here, this is the recombinant DNA. All right, this is the recombinant. This is the recombinant DNA. All right, this is the recombinant DNA. Now in this particular scenario of recombinant DNA, now once we have created this recombinant DNA, we create multiple copies of it. We basically do a photocopy, you can say. We do a photocopy of it, and we create multiple copies out of this recombinant DNA. And once we have created multiple copies, this will be inserted inside the plant, and it and the plant may respond to this, and the plant may accept the new genes, and this is where it starts to show the properties which are required. All the desired properties that we wanted out of the gene, now it will start showing up in this particular plant. So this is how this entire process works. All right, so first you will pick the gene of interest. Once we have picked the gene of interest, it will be inserted inside a vector. So we will choose the vector accordingly. Once we have chosen the vector, we will make the required cut in this vector, and then we will insert it inside. Once we have inserted it inside the vector, this is where you will now stick the ends together and create multiple copies of it. And once we have created the multiple copies, this is when now it will be inserted inside the required plant. So this is the entire process that works in recombinant DNA technology. So first, it is the isolation of the genetic material. So this is where we see that we will first isolate the desired DNA. We will isolate the desired DNA in its pure form. And once we have done that, we will use the restriction enzymes in ensuring that we determine the location at which the desired gene has to be inserted. And once we have done that, we use the ligation method, where we are ligation means joining, sticking the ends together. So with the help of the enzyme called as DNA ligase, so with the help of DNA ligase, we'll make the ends to stick together. Once we have done that, this is where we will create multiple copies of it. And this creating multiple copies is called as polymerase chain reaction, or PCR. So PCR is nothing but creating multiple copies of this particular recombinant gene. And once we have done that, this is finally where we insert the recombinant DNA into the recipient host. And even this has a name called as transformation. So ultimately, transformation means that you have now inserted the required gene into this particular plant. So this is the entire process. All right, this is the entire process that we are making the use of in creating any transgenic plant. Read this question. I'm launching the poll for you. Hmm. So 47% of you have marked D, 24% A, and 21% C. Okay. Recombinant DNA technology or genetic engineering allows the genes to be transferred across different species of plants. Can we do that? Yes, we can. From animals to plants? Why can't we do it? We can. What did we discuss? Let me just go back. Please pay attention. What did we discuss? That transgene can be taken from any other plant, right? It will be taken from some other organism. Which other organism we have not specified. We are not saying that it will only be taken from plants or only be taken from animals or only from microorganisms. We already have seen an example of microorganism where in BT cotton you are taking it from a microorganism. In case of golden rice, you are taking it from a plant. So theoretically you can take it from anywhere. That's why I told you that you take it from an alien. You find an alien on Mars, you bring it back and you take the genes and you insert it in the plant. There is no restriction put as such that which genes can be used and which genes cannot be used. Yes, some genes may give you better results than others. There are higher chances that a plant gene will give you a better result as compared to an animal gene, but it's not that you will not get something out of it, right? You will get something out of it. So please uh, ensure that you understand the things conceptually, that here we are saying that allows the genes to be transferred. It allows the genes to be transferred from anywhere, from any organism. Any organism. So that's why please don't confuse here. You can take it from any organism. That's why all the three are correct, and that's why answer will be D. Then there are other applications of biotechnology. For example, in case of biofuels also, we can call it as an application of biotechnology. Because in biofuels, what happens? There also we discussed that we are using crop plants, right? We are using crops and we are using seeds, etc. from these plants. And that is where you are getting your uh, biofuel from. So that's why in all these cases also we understand that uh, it is also an agricultural application of biotechnology. Similarly, we have something called as biofortification. Now, in case of biofortification, discussing about we are discussing about how exactly we can fortify the foods. Now, when it comes to biofortified foods, just understand that when we are saying that these are biofortified crops or biofortified foods, it means that we are enhancing something in that crop. All right, we are enhancing something in that crop. So, for example, let's say it could be enhancing some vitamin, or it could be enhancing some mineral. So it can be enhancement of anything as such. So overall, that's why we are looking at enhancement of something in this particular scenario. Now, when it comes to the enhancement, generally it is enhancement of micronutrients. All right, we are producing micronutrients or micronutrient dense crops. So when we say micronutrient dense crops, what it means is that uh, we are improving the amount of micronutrients in a certain plant. Now when we say micronutrient, what is micronutrient? What is the meaning of micronutrient? Any idea? Anyone who can tell me? Yes, vitamins and minerals. Why are they called as micronutrients? Yes, Vaishnavi, you have given the correct answer that they are needed in small amounts. So micronutrients, they are needed in small amounts. Needed in small amounts, and that's why they are called as micronutrients. So examples are vitamins and minerals. Vitamins and minerals, they can be called as micronutrients because they are required in smaller quantities as compared to carbohydrates or fats, etc., which are required in larger quantities in our body. So that's why they are called as macronutrients. They are called as macronutrients. And why macronutrients? Because they are required in larger quantities. Required in larger quantities. So that's why carbohydrates or fats, etc., they can be called as macronutrients. So that's why if you go to a dietitian, they would say macros. That if let's say somebody is overweight and they go to a dietitian, they would be said that you have to cut your carbs and you have to decrease your macros. So that is what they mean. Macros means macronutrients and carbs means carbohydrates. So that is basically how we look at micro and macronutrients. So generally what happens is that our body gets enough of carbohydrates, fats, etc. easily. But that's not the case with vitamins and minerals. There are sources of vitamins and minerals, and we need to ensure that we have a balance in our intake. Otherwise, it is in some way or the other, we might also become deficient. So that's why we see that there are all these things that have to be taken care of. So that's where fortification has to be done. So fortification will be addition of an ingredient to the food to increase the concentration of a particular element. So that's why in case of, for example, golden rice is an example of what? Fortification, biofortification. Similarly, iodized salt, it's not a biofortification because there is no biological source, but chemically you are adding iodine to it, so it's fortification. So that's why iodized salt is an example, golden rice is an example. So there are these examples of fortification. Similarly, sometimes we use the process called as supplementation. The meaning of supplementation is that instead of taking that micronutrient through your food, you are taking it through a supplement. All right. So, for example, vitamin D capsules or Bicosfuls or vitamin B12 capsules, etc. They are what? They are supplements. So that is called as supplementation. All right. It is called as supplementation. So addition of an element to the diet to make up for an insufficiency. Then there is something, a concept called as hidden hunger, a concept that has been defined by WHO, so that they call it as hidden hunger. Now, when it comes to the concept of hidden hunger, this is where what they say is that although you are eating enough, you are not getting all the nutrients that are required for a healthy functioning from of your body. So in a manner, we are talking about an undernutrition in an individual that happens where maybe they are receiving sufficient energy, they are receiving sufficient carbohydrates and fats, but they are not getting essential micronutrients in a particular amount, in the required amount. So that's why in this particular scenario, they would be called as, that scenario will be called as hidden hunger. Now, So hidden hunger basically you can say is that this is where you are talking about a form of undernutrition, form of undernutrition when a person gets enough energy but not enough essential micronutrients. Let me write micronutrients. Essential micronutrients. Now, this is a scenario, especially in the countries where uh, they are dependent on a lot of food imports, where they are not capable enough with their agriculture and they are dependent on some other countries so that they can supply the food. Now, whenever food supplies happen, they are largely, you can say, carbohydrate based: rice, wheat, pulses, etc. All these things will be provided. So, you get enough of macronutrients, enough of uh, carbohydrates and fats and proteins, but not enough of vitamins, not enough of minerals. So that's why that becomes a form of hidden hunger. All right, this is what we call as hidden hunger. So today we see that there are a lot of biofortified crops that are being used today, and overall we have been making the use of all these to supplement or to complement all the things that we are eating. So for example, let's say cassava. So in case of cassava also, what we see is that sub, fortification of vitamin A is essentially done in case of cassava. Similarly, we have maize. Now, in case of maize, also we see that there are certain fortifications that we do, and overall, especially when it comes to maize, we see that deficiencies of proteins actually are largely taken care of with the help of fortification in maize. So here this is one example where not only not only the micronutrients but even macronutrient supplementation is also being done. Similarly, we also have sweet potato. Now, in case of sweet potato, what we see is that here also we do supplementation largely of vitamin A. So largely what we see is that in this case also supplementation of vitamin A can be done. Then when it comes to beans, in case of beans, what we see is that this is where we will be doing supplementation largely of iron. So iron based supplementation is done in this particular scenario. Then after that, when it comes to millet, in case of millet also we see that iron deficiency is taken care of. So iron also, and in certain cases zinc deficiencies are also taken care of with the help of millet. So there also we see it. Then similarly, when it comes to uh, rice and wheat, obviously vitamin A is done, and also zinc supplementation is done in this case as well. So fortification, not supplementation. So that's why there are all these examples. In every example we see that in one way or the other we are taking uh, we are making the use of biofortified crops, and that's why we are making ensuring that any kind of deficiencies, especially in the case of micronutrients, can be taken care of. So that's why this is how we look at the various aspects of it. Similarly, genetically modified crops as animal feed are also available now, where we see that overall a lot of things can be taken care of with the help of these crops. That uh, instead of feeding the animals a lot and not providing them with enough nutrients, what we want is that with smaller quantities, if we can enhance the amount of nutrients that we can get there. So that's why we see that this is something that is doable. Similarly, pest resistant cultivars, meaning that we are, for example, we discussed about bt or bacillus thuringiensis. Now in this case also we see that this is where we are using certain bacteria to enhance the quality of crops. So BT cotton becomes an example where we are making pest resistant cultivars. Similarly, we are also making genetically engineered herbicide tolerant crops. All right. herbicide tolerant crops. Now, in case of herbicide tolerant crops, what we understand is that these are HT variants, meaning that they will not have a negative impact because of herbicides. Now, herbicides will be used to kill any unwanted growth, all right, any weed that is growing in any crop plant, that for weed control we make the use of herbicides. Now at the same time, herbicides might also have a negative impact on the crop itself, all right, on the crop itself, and that's why we don't want that. So we build HT variant crops, where the HT variant crops will be able to withstand the herbicides, but the herbicides will kill all the weeds that are growing in the crop. So overall you will also be able to improve the efficiency of the growth of the crop. So that also is a very, very important factor here. Then the use of biofertilizers. Now, in case of biofertilizers, also we see that this is where we discussed about this in biogas plants also, that you have the leftovers which can be used as biofertilizers. So biofertilizers also can be called as a product of biotechnology, where we are creating these kind of fertilizers which are biological in nature, and hence they have less negative impact on the soil, and they would be able to enrich the quality of the soil without having a negative impact, without having a poison being mixed in the soil or the nearby water bodies. So that's why we see that this is also one place where they would be able to do well. And then improvement in floriculture, that even in the flowers, and it's that's why we have to understand that it's not only about the food, or not only about the food crops, but even in the commercial crops we'll be able to do a lot better. For example, when we talk about floriculture, it means that this is where we'll be able to improve in floriculture, we'll be able to improve upon in floriculture, and by improving in floriculture we'll be able to get better output in every sense, whether it is about color or scent or size or the long life of a flower. All these can be enhanced, all these properties can be enhanced in a flower, and that's why improvement in floriculture can also be seen with the help of biotechnology. So that's why there are multiple different ways in which biotechnology helps in agriculture, and today we have been focusing upon a lot of different ways in which agriculture can be agriculture can be helped with uh, biotechnology. So another question, if you see this, this was asked in your 2011 paper. I launched the poll. Okay, you tell me the answer. I'll not launch the poll for this question. You tell me what you think is the answer. Can you tell me in the comments? Hmm, it's interesting. So a lot of C's and a lot of D's. All right. So let me discuss this with you. Other than resistance to pests, what are the prospects for which genetically engineered plants have been created? Okay, so some of you have said that. Okay, let me discuss this. So first says to enable them to withstand drought. This is doable, and we already have been doing it. That we have created plants, a lot of genetically modified plants, which can grow in areas where there is a water scarcity. So even with less water, they can withstand these kind of areas. So yes, that's possible. We have already done that. Then uh, to increase the nutritive value of produce, this absolutely is possible. We discussed about golden rice and so many biofortified crops. So even doing this is possible. S- Statement 3 is to enable them to grow and do photosynthesis in spaceships and space stations. Now, is that possible? Today, yes, it is possible. Today it is actually possible, and we have seen that there have been certain programs that have been launched by NASA in the past few years, where they have actually done this. So in 2016 they had done an experiment, 2019 they did an experiment, 2022 they did an experiment. So there have been all these experiments that they have conducted over the years, where they have showcased that how exactly doing this is actually possible. So now it is possible, but this question was asked back in 2011, when until then it was never done. Now it has been done, that there is a veggie plant growth system, and they have used that veggie plant growth system to do it. Similarly, they have advanced plant habitat, so that also has been a program where they have done it. So there are now these programs where NASA has shown this, and in International Space Station we have done it. But this question was asked in 2011, and look at the wordings of this question. The question says what? Other than resistance to pests, what are the prospects for which genetically engineered plants have been created? Now, have been means what? Have been means that they should have been done already. Past continuous tense. So they should have been done already, and that's then only you can mark it as correct. Let's say the question was can be created. If they had asked can be created, then can be opens up possibilities. So then you can close your eyes and without even reading anything, you can mark all the four as correct. But that's not the case here, correct? That's not the case. They have not asked can be, they have asked have been. And with have been, you have to have an example, you have to have examples of the past, then only you can mark it as correct. So that's why 3 could not have been marked correct in 2011, but it could be marked as correct in 2024. And then fourth statement is to increase their shelf life. We already discussed this, that it is a desirable property that we have been doing. So that's why the correct answer in 2011 was C, and the correct answer today is D. So this is how the things have changed. So that's why I did not give you the poll. Otherwise, everyone who marked C, if I marked D, you would be angry with me, and everyone who marked D, if I marked C, you would be angry with me. So that's why I took uh, a mid step. Clear? So any question if you have from whatever we have discussed until now? Is there any extra session you'll be taking as it's showing in the dashboard? Extra session? No, there has been a cancelled session, so I'll make up for that cancelled session whenever I can get time. Are proteins part of macronutrients? Yes, they are. I got only 70 to 80 marks in mock. What do I do to increase my marks? See, a couple of things. First, getting 70 to 80 in mocks is above average, I would say. It's good enough because a lot of questions in the mock test that we give are difficult questions. So that's why somebody getting 70 to 80 in the mock examinations, I would say you are doing good. But obviously, you should try to increase it. How do you do that? Look at the leftovers, that what you could not score. Either you left that question because you did not know the answer, or you marked it but it was wrong. Whatever you left it because you did not know the answer, right? So now go back to those questions, look at the solutions, look at the analysis, and then start going back to that chapter. That is it related to something that you studied and could not answer, or is it something related to that you have never heard of and that's why you could not answer? Now if you have never heard of it, that's fine. You read it there and ensure that it becomes a part of your learning for the future. But if it is from a portion that you have done in the class, yet you did not remember it, then you have to go back to that portion of that particular chapter and ensure that you revise that chapter well, and especially that portion in the chapter well. That, for example, let's say it was related to biotechnology, but especially the questions related to agricultural applications of biotechnology, you could not answer well. So it means that this is one portion in that chapter where you are weak. So you have to go back to the board and do a revision of that. So that's why you first have to analyze that where did you go wrong. Was it because you marked a lot of wrong answers, or was it because you did not know answers and you could not mark it? So based on that, you have to strategize in a manner that you learn out of it, that whatever you left out or whatever you committed mistakes with, you do not repeat those mistakes in the future if something similar repeats in the examination. Why is there a protest against BT cotton? Now, what has happened with BT cotton of late is that the BT cotton is not showing the same results as it used to. There is a wonderful article that has been published in the Down to Earth magazine also on this, where the article has published that how again the productivity has started going down, and bollworm is able to affect BT cotton. Bollworm also has evolved in a manner that it can feed upon BT cotton now. So that's why now we have seen that there is a decline in productivity of BT cotton, and that's why the farmers are worried, and a lot of people who are associated with the entire agriculture of cotton, they are worried. So that is where the problems have now started. If you can, please go to Down to Earth and just type BT cotton Down to Earth on Google. You will get that article. It's a wonderful article that gives you insights on what has been happening off late with BT cotton. Please inform in class before taking extra class. I'll do that. I'll check with the team when exactly is the extra class scheduled, and I'll inform it duly in the classroom beforehand. So don't mind. Can we make hair transplantation through genes? I don't mind. I have accepted the reality, but yes, doing, see, right now there are only few things that work, and hair transplant is something that has worked for many, and that is one of the possible solutions right now. But yes, there are therapies that are also being tried. There are a lot of novel therapies. For example, stem cell therapy is being tried in these kind of scenarios. So a lot of these kind of things. But see, when it comes to genetic treatment, largely genetic treatment is often seen as one of the solutions where you are looking at diseases, when you're looking at life threatening diseases where you need something because you do not have a medical therapy. But something which is a cosmetic therapy is never, you can say, the first priority. So that's why we see that there are developments in that field, in genetic engineering as well as in stem cell therapy, but yet not a lot of success has been seen with it. Do we have a class tomorrow? I'll check with the team during the break. I'll check it and I'll inform you after the break. PCR represents the whole DNA combinant process again and again, or some type of regeneration from ligase DNA. Basically, PCR would be that recombinant DNA that we have produced that has an extra DNA. We basically produce photocopies of that particular gene. All right. So that particular gene we create multiple copies of. Can we read class 12th bio book for this section and disease section? Please avoid it. Not required, because there are a lot of things which are technological in nature and we don't need to go to that level. Whatever was needed, I've already taken out some of the information from that, uh, those two chapters of biotechnology that we have right there. No need to go beyond that. And also with regard to diseases, uh, a lot of information unfortunately is outdated as well. So I would still prefer that you don't go through that, and it's very technical in nature, not meant for an examination like civil services. Here a lot of nutrition related things were there. Then why some people use chemicals to grow earlier crops? See, In agriculture, we have started finding these ways only off late. And the success rate has not been that great. Using chemical fertilizers has been much easier, much cheaper as well at the same time, and with a lot of good productivity. So that's why we have been looking only towards that productivity factor, forgetting about the other aspects of how it can negatively impact human health, how it can negatively impact the environment and other organisms as well. So that's why we have seen that we have been only focusing on productivity, that has led to a lot of chemical pesticides and fertilizers being the primary source in agriculture. From where these magazines can will find in our areas, it is not found. It's not about getting the print version of the magazine. Online versions of the magazines are also there. Either you can take subscriptions, or there are other ways to get it. And also many of these articles will be there on their website also. So not necessarily that you need that article always, or you need that uh, magazine always. And also we keep on analyzing those magazines, and we keep taking the important articles out of those magazines all the time. So we also help you in that sense. Any previous year questions will be repeated again? Is there any chance? Hasn't happened a lot. There is only one question from ancient history, from Buddhism chapter, that incidentally repeated, and 2 years in a row a question actually was asked from there. So I remember only one of this example where it has actually happened. But apart from that, words change, some of the areas could be uh, the same. It's a possibility, but overall the same question coming twice, highly unlikely. One pair of chromosomes is different among 22. If you are talking about the difference between males and females, that is where the 23rd pair is different. List of radioactive elements and uses not shared yet. I'll check that during the break. Spider-Man movie is based on DNA recombinant process, hypothetically. Yes, of course it is. Many have tried this recombinant DNA technology in their homes, especially during childhood. Unfortunately, hasn't worked for one of us. Which online sites can we access for this portion? Uh, in what context are you asking? Can you let me know? Are nutrient tablets made through gene modification? Not really. In many of the cases, they actually are made of chemicals, and they are derived from these chemicals only, and there is nothing genetic in it. Janani Suraksha this year also from 2011. Yes, true. It did repeat. Where chromosomes are present in our body, every cell, every cell of your body has nucleus, and every nucleus has chromosomes. Hmm. So let's move on to the next topic. And now we have white biotechnology. Now, in case of white biotechnology, we basically are talking about industrial applications, that how in the industries we can make the use of biotechnology. Now, here also there are various different ways and various methods that we can use. So, for example, the process of fermentation. The process of fermentation is where we can make the use of white biotechnology. Similarly, when it comes to oil recovery, in oil recovery also microorganisms are used. microorganisms will be sent to the oil wells, and these microorganisms will eat up all the hydrocarbon based fuel, and then from these microorganisms we derive these hydrocarbons. So that's why in oil recovery also we make their use, and then they can also be used as preservatives. All right, they can also be used as preservatives, so that also becomes a very very crucial way in which it can be useful. So that's why we see that there are different ways in which we can make the use of biotechnology in industries. Similarly, we discussed about biofuels. Even biofuels can be considered as an industrial process also. So that's why, although it is green biotechnology also, we can consider it as white biotechnology as well. Now, then there are some of the other processes such as biosorption. Now, when we say biosorption, in a manner, you can say that there are two terms that are combined here: biological absorption. So, biological plus absorption becomes biosorption. Now, what are we doing here? Certain types of inactive dead microbial biomass, they bind and concentrate heavy metals from even very dilute aqueous solution, meaning that even from the environments where you have very small amounts of something present, from there also you can extract something. So especially in the extraction of heavy metals, in many of the processes biosorption can become a way in which we can do that. So again, the same microorganisms will be used, and with the help of these microorganisms we can try this. So this is where we see that microbial biomass can be used to bind and concentrate, because they'll not be able to break down. All right, just understand this, that they will not be able to break down these heavy metals. They can extract these heavy metals, and then we can remove these heavy metals. So it is not like the normal process where these microorganisms or decomposers, they're able to break down the substance, they're able to break down the material. But here in this case, they're not going to break down the material, but rather what they are going to do is that they are going to extract it out of the soil, or extract it out of the water, or maybe out of the industrial units. So by doing that, they basically take care of any contamination, etc., that might be there in a given area. So even from very dilute aqueous solutions also we can make the use of microorganisms and their use to extract these heavy metals. So that's why this is also something that can be done. Similarly, there is something called as biomining. So again, mining with the help of biological resources. That's why biomining. That's why this is called as biomining. So why biomining? Because this is with the help of biological resources that we are doing it. So overall, when it comes to biomining, we see that this becomes a process for extraction of desirable minerals from the host with the help of living organisms. Remember, living organisms, any kind of living organisms. Largely, we are making the use of microorganisms, but we can also make the use of plants for biomining. So, for example, microorganisms can go into the oil wells, and from there on, we can extract them. And especially for the dilute environments, we will prefer the use of microorganisms. All right, we'll prefer the use of microorganisms from the oil wells, because in dilute solutions or dilute environments, because if you use the normal industrial methods, the energy spent will be higher than the energy that you are extracting from there. So that's why those methods may not be feasible in these cases. Microorganisms can become a better way to extract energy out of it. Similarly, when it comes to plants being used, it is called as phytomining. So when we say phyto in general, whenever you find this term phyto, it means plants. So wherever plants are being used, this is where we call it as phytomining. So phytomining, that's why, for example, let's say the roots of certain plants. So roots of the plants can extract metals. For example, for copper extraction, we make the use of plants. The roots of the plants will extract copper, and then we'll take the copper away. We burn the roots, and from the ashes, we collect copper ions from the ashes. So in this manner, we see that plants also help in biomining. So that also becomes one of the ways in which we can make the use of biomining. Similarly, there is something called as blue biotechnology. Now, in case of blue biotechnology, we are making the use of biotechnology, or in case of aquatic solutions, in the aquatic environment. So that's why it is called as blue biotechnology. So when it can be used to develop microorganisms to clean water bodies, just like oil spills. Now, how exactly we do that? Whenever oil spills happen on any sea surface, any ocean surface, wherever oil spill has happened, we take something which is called as oil zapper. All right, we take something called as oil zapper, and this oil zapper will be introduced to this area wherever oil spill has happened. Now, this oil zapper is a formulation made of five different types of bacteria. All right, you have five different types of bacteria, and you've used this by bacterial formulation for your for treating the problem. So what happens is that as oil zapper is dropped on this ocean surface or the sea surface, it simply goes and starts eating up the hydrocarbons and breaking down the hydrocarbons. So these hydrocarbons will be broken down into carbon dioxide and moisture. Hydrocarbons will be broken down completely. They do not just digest them, they simply would break them down. So all the hydrocarbons which are there because of the oil spill, they will be broken down into carbon dioxide and water, and by doing that, oil zapper will take care of whatever oil spill might have happened. So that's why by using oil zapper, we can take care of the oil spills that might have happened on the ocean or the sea surfaces. So that's why it's a mixture of five types of bacteria which feed upon the hydrocarbons, and they convert the hydrocarbons into carbon dioxide and water or moisture. So in this case, we see that this is how biotechnology can help in creating transgenic aquatic organisms also with desired properties. So in this way, we see that there are a lot of ways in which we can make the use of blue biotechnology as well. Then after that, there is a very important process where we make the use of biotechnology, and this is called as bioremediation. Now, bioremediation is when we are making the use of biotechnology in the environment. When we are making the use in the environment, we call it as bioremediation. Now, bioremediation, that's why it is the cleaning of the environment with the help of living organisms. With the help of living organisms, cleaning of the environment. Now, in this particular scenario, when it comes to these living organisms, we are talking about the living organisms that in one way or the other can help in cleaning the environment. There are two processes that we can use here. One which is called as in situ bioremediation, and the other one which is called as ex situ bioremediation. So understand what do we mean by in situ and ex situ. Now, when we say in situ, the meaning of in situ is at the site. The meaning of in situ is at the site, and meaning of ex situ is away from the site. So in situ means at the site, and ex situ means away from the site. Meaning that when we are doing the bioremediation or cleaning up of the environment at the site of contamination itself, then it is called as in situ bioremediation. Similarly, when we are doing this away from the site, all right, when we are doing it away from the site, then in that case it is called as ex situ bioremediation. So that's the basic difference between in situ versus ex situ. In situ is at the site of contamination, ex situ is away from the site of contamination. So this is how we look at the various strategies that are required for bioremediation. So now to begin with, the first one is in situ bioremediation. Now in case of in situ bioremediation, there are various techniques that we use, and all these techniques basically do the same thing, that they are doing all this activity at the site of contamination itself. They are doing the decontamination activity at the site of contamination, and that's why they are called as in situ bioremediation. So there are various techniques that are being used. There is biosparging, bioventing, bioaugmentation, biostimulation, there is bioculture. So there are all these different methods that are used in situ bioremediation technique. Now, when it comes to all these techniques and when it comes to all these bioremediation techniques, understand how exactly they work. First one is biosparging. Now, biosparging as well as bioventing. These two are very similar processes. All right, these are very similar processes. So just try to understand how they are similar and where is the difference between the two. So where exactly do we use them? So let's say we are talking about a layer of soil, right? We are talking about a layer of soil. So you have this layer of soil, and below the layer of soil, you might have the water table, right? You might have the water table. So you have the water table below the soil layer, and above that, you have the soil layer itself. Now, in both these layers, in both these layers, the microorganisms will already be present. The microorganisms are already present, correct? And they are naturally working in biodegradation, right? They are helping in bioremediation naturally as well. So it's not that they are not doing the process of degradation. They are working in the process of biodegradation, but that process is being carried out very slowly. All right, that process is being carried out very slowly, and that's why it's not enough. The entire process or that speed is not enough, and we need to gain that speed. We need to gain that speed. So to gain that speed, what do? We know that they are working very slow. We want them to work fast. We want them to do the degradation process, the biodegradation process faster. What do we do to ensure that? We feed them. We feed them, we give them food, we give them nutrients, we give them oxygen, and in the presence of that extra food and oxygen, they would be able to start working faster. Now they get their nutrients, and as they get their nutrients, they get that energy to start working faster. And exactly this is what they do, that they start working faster. And now, when we use this process, when we are using this process in the so-called saturated zone, it is called as biosparging. And when the same process is being carried out in the unsaturated zone, it is called as bioventing. Now, what do we mean by saturated and unsaturated? Now, saturated zone is this zone. This one is called as saturated zone, and this zone is called as unsaturated zone. Okay, this zone is called as unsaturated zone, and the reason is simple. Why saturated? Because it is saturated with moisture. It has enough of water. It cannot sustain extra moisture apart from what it already has. So that's why it is saturated. And unsaturated because it still has the capacity to engulf more water, to absorb more water, and that's why it is unsaturated zone. So when this process is being carried out, basically giving the nutrients and oxygen. So when we are giving the nutrients and oxygen to the saturated zone, when the nutrients and oxygen is being given to the saturated zone, then in that case it is called as bioventing. Sorry, sparging. And when it is being given to this unsaturated zone, it is called as bioventing. So this is bioventing. This is bioventing, and this is biosparging. So this is biosparging and the other one is bioventing. So bioventing, that's why it can be done at low temperature, sorry, low pressure. It can be done at low pressure, and this is something that is done at high pressure. Now this is also simple to understand, that why high pressure and why low pressure. High pressure because you are going deep underground. So that's why you need more pressure so that you can send the required oxygen and nutrients to the required level. But in case of bioventing, you're not going deep down below. You're only going to the above part. You're only going to the part that is easier to reach. And that's why here it can be done at low pressure as well. So that's why bioventing will be done at low pressure and biosparging will be done at high pressure. So in biosparging, air or oxygen and nutrients are injected at high pressure to increase the biological activity of the indigenous microorganisms and enhance their decomposition activity. Similarly, in case of bioventing, what we do is that here uh, we are also introducing the same air oxygen but at low pressure this time, and here we had done the same at high pressure. So the processes individually, you can say that these are the same processes, but just that one is being done in the upper part of the soil and the other one is being done uh, below the water table, and that's why one can be done at low pressure, the other one has to be done at high pressure. Individually the same things are being done, that you already have the microorganisms and we just want to enhance the activities of the microorganisms, and to enhance these activities we are providing them with more food, and that food being provided they get that extra energy and they start working faster. So this is exactly what we are doing in both biosparging and bioventing. So both these, that's why just remember, and these are processes that are very often used in these cases, so that's why they become important. Apart from that, there are other processes also that are used many a times. Now for example, we discussed we have seen other another set of words here, like you have bio augmentation, biostimulation, etc. as well. So bio augmentation, what we do is that we still do not move away from the contamination, but this time we bring the microorganisms to the site. In biosparging and bioventing, the microorganisms were already present, just that we wanted to enhance their activities, right? We just wanted to enhance their activities, but this time we do not have these microorganisms, we have to bring these microorganisms to the site of contamination, something that we just discussed 5-10 minutes back. Can you tell me one example that we had discussed, one example that we had discussed where microorganisms were being brought to the site of contamination? What was that? Yes, Kushi, you are correct. Yes, Bala, you are correct. Kumar, you are correct. We discussed about oil zapper, right? We discussed about oil zapper, and what did we discuss about oil zapper? That now you have to bring the microorganism to the required site. So that is what augmentation is, that here we bring the microorganisms to the site of contamination. We bring the microorganisms to the site of contamination. So this is exactly what we do in bio augmentation. So overall, this is basically how we make the use of bio augmentation. Similarly, there are other activities. For example, we can make the use of biostimulation. So biostimulation is when we are trying to just enhance the rate, right? When we are just, this is about enhancing the rate, enhancing the rate of bioremediation. Enhancing the rate of bioremediation, especially the contaminated soil and water, can be done with the help of this. So there are multiple different ways in which we can do it. There are electron transfers that we do, pH adjustments that we do. So there are multiple different ways. Temperature control can be another method, and even biosparging and bioventing, in a manner you can say, are in a manner they are also similar to biostimulation. So we enhance the rate of bioremediation in any way possible in biostimulation. So there are all these ways that we can use in situ. Then we have ex situ bioremediation techniques. So in ex situ techniques, there is first one that is called as land farming, where you are doing nothing else but just you are plowing the fields in a manner, right? So periodically tillage of the soil will be done, and with the tillage, what do? You are basically trying to wake up the microorganisms. So for that, first we take up the soil away from the site, will take up the soil in some kind of area, and their periodic tillage of the soil will be done, and that tillage of the soil in a manner wakes up the microorganisms, ensuring that they will start with their activities, they'll start working on their activities faster. So that's why this is called as land farming. So we contaminated soil basically spread over a prepared bed, and soil is periodically tilled to stimulate the growth of microorganisms for degradation of organic waste. Then there is something called as biopile. Now, in case of biopile, what we are doing is that in case of biopile, we can say that it's a hybrid technique of land farming and land composting, where what we do is that we take the required soil, and as you can see in this picture, this soil will be taken to some facilities, and that is where we take utmost care of that soil. Every parameter, every aspect of the soil will be well maintained. What temperature is required, what pH levels are required, what kind of moisture is required, what nutrients are required, so the kind of heat that you need here, all these things will be well controlled, and the soil will be kept here for a long time. Even a lot of, let's say, uh, biodegradators, uh, they will also be used. Earth forms can be used, all these will be used, and they also are natural, uh, you can say, ways in which they can enhance the quality of the soil, and it can take up to 3 to 6 months of time to enhance the quality of the soil as per our requirement. So that will be called as biopile. So moisture, heat, nutrients, oxygen, pH, all these things will be controlled, and typically it can take up to 3 to 6 months, after which we see that the required soil can either be sold or can be returned to the original location. So this is how the treatment will be done, and will ensure a much better quality with this soil. Then apart from that, there are some other techniques as well. So for example, we have phytoremediation. Now I told you what phyto means, that wherever you are making the use of plants, if plants are being used for remediation techniques, then we'll call it as phytoremediation, right? We'll call it as phytoremediation if plants are being used. So phytoremediation, now we know that all the plants anyway help us in remediation. All the plants take in carbon dioxide, then they give you oxygen, so they help in cleaning of the environment anyway, cleaning of the air anyway. But apart from that, there are other ways in which they can help. For example, if you consider a plant like neem, we know that all the different parts of the plant, all the different plants of this neem tree can in one way or the other can be useful for us. So its bark can be useful, its seed can be useful, its leaf can be useful, its flower can be useful. They all can be useful in one way or the other. So you can say that in a manner it is a complete package that helps in bioremediation, and also a lot of other ways in which they can have health related benefits as well. Then there is something which is called as phyto extraction. Now, when we say phyto extraction, extraction means what? That you are extracting something, you are pulling out something. So, what exactly are they extracting? Anything that cannot be uh, cleaned up with the help of microorganisms. For example, heavy metals. We discussed about heavy metals, that microorganisms cannot degrade it, but rather they can just eat it up. So, similarly, in case of phyto extraction, what we do is that the roots of the plants, they will extract all the heavy metals, they will extract compounds and elements from the soil and water, and in this way they would be able to extract everything from the soil. So they would be able to extract the contamination out of the soil. So these roots that you have, these roots become very very crucial in that sense. They would be able to clean up the soil and make the soil more nutritive and more productive altogether. So this will be called as phyto extraction. Then after that, we also have techniques such as mycoremediation. Now the term myco is the use of fungus, and as you call in plural terms, fungi or fungi or fungi or whatever you want to call, as long as you write the correct spelling, that is fine. So that's why, to be on the safer side, I've used the word fungus. So mycoremediation is the involvement of fungus, and where we are making the use of fungus in bioremediation. Now a lot of them, they also can be used in enhancing the quality of the soil. They also are the ones that can help in fixation of the nitrates in the soil, and hence they become very very useful in that perspective. So that's why we see that there are a lot of ways in which all these different microorganisms and plants, etc., they can help in bioremediation in one way or the other. So that's why we see that this is how all these things can be done. Read this question. This also has been asked in your paper. I'll launch the poll for you. Hmm. So I see that 44% of you have marked C, which actually is the correct answer, and I'll tell you why. In the context of solving pollution problems, what is or are the advantage or advantages of bioremediation technique? First, it is a technique for cleaning up pollution by enhancing the same biodegradation process that occurs in nature. We discussed about that exactly what we were trying in biosparging and bioventing, that naturally also degradation would have happened, but we wanted to enhance the rates, and hence we added extra oxygen, hence we added extra nutrients. So that's why this is correct. This is what we do. Second statement is any contaminant with heavy metals such as cadmium and lead can be readily and completely treated by bioremediation using microorganisms. Now, there are two problems with this sentence. One, we are saying that we will treat heavy metals with the help of microorganisms. What we discussed is that for heavy metals, we would rather make the use of plants, the phytoextraction techniques. Second, another problem that we have with this one statement is, look at how they have given this entire statement. Any contaminant with heavy metals such as cadmium and lead can be readily and completely be treated by bioremediation using microorganisms. Now, this is where what we call these as extreme words. These are all extreme words, where if you see these kind of extreme words in any of your statements, you should at least read that statement twice or thrice, and you'll find that in majority of the cases these kind of statements are wrong statements. And precisely that's the case here as well, that we cannot say readily and completely. What we said is that they can barely extract these metals, they cannot degrade this metal, right? So that's why we cannot really say that it can be treated by bioremediation using microorganisms readily and completely. So that is where it becomes very extreme, and that's why this is wrong. All right, that's why this is wrong. Third is, genetic engineering can be used to create microorganisms specifically designed for bioremediation. This is also correct, and a simple example is oil zapper, where we are making the use of genetic engineering to create a formulation out of five different types of bacteria. Similarly, there is also something called as bioculture. In bioculture also we do something similar. So all these are possibilities. So correct answer, that's why will be C, that is only 1 and 3. Read this question, a very simple question. I'm launching the poll. This is simple, I need more people to answer this correctly. Good. 73% of you have marked it correct. The correct answer will be A. So, oil zapper, we discussed it in the context of oil spills. So, that's why this is correct. A is the correct answer. Latest technology developed for undersea oil exploration. It's not about oil exploration, it is about controlling the oil spill on the surface, right? On the surface of the ocean and the seas. So, it's not about undersea exploration. So, this is wrong. Genetically engineered high biofuel yielding maize variety, completely wrong. Latest technology to control accidentally caused flames from ocean wells, again nothing to do with ocean wells. So correct answer, that's why will be A. All right, so any question from this segment? Any question from this segment? List of extreme words. List of extreme words, there are many. I mean, for example, any or none, always, never, or completely, readily, as we saw here. All these kind of words, they can be said to be extreme words. Is biostimulation similar to biosparging and bioventing, as in all these three cases we try to enhance the remediation process? See, you can, in a manner, what you can say is that biosparging and bioventing both are a kind of biostimulation. Both of them are a kind of biostimulation. There can be various different types of biostimulation, and biosparging and bioventing are two of the examples of biostimulation. Which plants are used in bioremediation? I mean, any big tree actually can be useful. Any big tree which has very deep roots, they all can be very useful. Once again, bioremediation. Bioremediation is nothing but just cleaning of the environment. Cleaning of the environment with the help of biological resources. That's why bioremediation. Lots of complex words, how to remember all of them? That I agree. I told you in the beginning of this class that this will happen in this, especially. And if you look at the previous year questions also, unfortunately this is how they have been asking questions. So that's why I've tried to put a logic to every word so that you can go one by one and try to understand how they are connected with the larger discussions that we are having. And that too, as I told you, that remembering them in one line, one sentence is more important than knowing the entire technology that goes behind the scenes. So that's why just do this much. It will be very helpful for you. It is difficult to remember all the types of bio. I understand the same thing. Both of you have written the same thing. If a person has been eating rice as staple diet for a long time, not much macronutrients, and after eating every day, not hungry for eating too much rice, is only, can we say hidden hunger? See, in that case also, it is kind of the same scenario. But hidden hunger, when WHO uses the word hidden hunger, they use it specifically in the cases where the people do not have a choice. Here what you have written is where you have a choice, but it's your dietary preference that is leading to that kind of a problem. But when WHO has mentioned hidden hunger, they have mentioned in the cases where the people do not have an option and they're just eating what they're being provided. So that is where the difference is. Otherwise this happens, and we see that this happens with a lot of people, that some kind of deficiency will start building up. For example, let's say for a lot of vegetarians, for example, uh, B12 deficiency can happen because a lot of non-vegetarian resources, they are good sources of vitamin B12, and that's why people who are vegetarian, they have to look into also the fact that the normal food that they are eating, uh, they that normal food does not have a lot of B12. So you have to ensure that you are eating enough of fruits, etc., which are good sources of vitamin B12. And by ensuring that you are getting enough of B12, uh, that otherwise you're not getting because of the vegetarian diet. So that can happen, and this starts developing in some vegetarians in with age. What is stunting? See, stunting and zero hunger is where people do not have the access to food at all. They do not have enough where for 24 hours they have not eaten, or for 24 hours they do not have the access to food at all. So that is where the problem starts, and it can lead to stunting over a period of time. Plant or animal which is difficult to get genetic material. Difficult to get is not the correct way to ask, but yes, genetic engineering is far more complex in animals as compared to plants. In plants the success rate has been higher as compared to that in animals, because genetic structure in animals is more complex as compared to plants. Any other application of bioremediation? There are many other. What I've taken is all the ones which are more common techniques and which are more used techniques. Otherwise the list is, I mean, on and on, and we cannot go into that much detail. Tomorrow class or not, I'll confirm this after the break. I'll confirm this. Hmm. So, can we take a break here? We have a next topic, that is cloning, which is a big topic. So let's not take a break midway. Instead, Let's take a break before this, and then we'll come back. So the time is 6:33. Let's take 15 minutes. Let's be back by 6:48. Don't take more. Don't make 48 as 50, please. Welcome back, everyone. So, what we have done until now is that we have discussed all the topics with regard to the various aspects of biotechnology, the various branches of biotechnology. Now, we move into some of the important topics which are crucial and which are very often in the news and in discussion in one way or the other. So, this is where we'll discuss about various topics that are in the news. For example, what is cloning? What do we mean by stem cells? What is DNA fingerprinting? What is GM mustard that is often in the news? What is the concept of three parents baby? So we'll discuss about all these kind of topics, which very often in one way or the other will keep coming in the news. So first is the concept of cloning. Now cloning is a process where we say that it is a process of asexual reproduction. We are saying that it's a asexual reproduction. Now, the meaning of asexual reproduction and the difference between sexual and asexual reproduction. In sexual reproduction, you would have a male and a female, and then the fertilization that happens, it happens between the male and the female cell, and then after that, the journey of an individual starts from there on. So that is sexual. And asexual reproduction is where only one parent is involved. There are not two parents. There is not a mother and a father. There is only one single parent, and that one single parent is leading to the birth of a new individual. That is called as asexual reproduction. For example, cell division. In a lot of microorganisms, we see that only what happens is that their nucleus will divide into two parts, and that will lead to the cell dividing into two parts. So that becomes cell division. Similarly, there are other techniques. For example, budding. So in budding, what happens is that a bud would appear in the body of the organism, and that bud starts growing, and after it grows it detaches, and once it detaches it becomes an own individual. So this also becomes a manner in which reproduction happens in these individuals. So this is called as asexual reproduction. Now in cloning also, we are saying that cloning is also asexual reproduction. Why? Because here also only one animal is involved, and what we are creating is an exact replica of that animal. We are creating an exact replica of that animal, and that is cloning. So cloning, in often we use that this term cloning also when we are trying to create a photocopy of something or an exact replica of something. We say that it is a process of cloning. So now this process of cloning is where we are, I'm saying that we are creating an exact replica, duplicates of animals or exact replicas of the animal. So the offspring or the progeny is an exact replica of the single parent who has contributed to the genetic material. Now, the process of cloning. If you have seen enough of films and TV shows, you might have seen that there will be a box, and one person will be sent inside a box, and there will be another box from where another person comes out. And in this manner, cloning might appear to be that simple, that there will be some mad scientist who just presses some buttons, and then one another person or an exact replica of the other person who went inside the box comes out. Unfortunately, things are not that simple. If you have seen Shaktiman, it happens in one of the episodes of Shaktiman. I don't know how many of you are here from the Shaktiman era, where we used to wait and watch Shaktiman. So there was one episode where a replica of Shaktiman was created by one of the mad scientists. Now, anyway, but uh, coming away from what TV shows and films have shown us, the process of cloning is very different. The process of cloning is very different, where we actually take the animal cell and we try to replicate it. And that process has been very unstable, very difficult to implement, but we got success. And this was the first success. I'm sure you know the name of this sheep. This is Dolly. The name of this sheep is Dolly, and this was the first ever case. This was the first case ever of a success in animal cloning that we ever got. And this was back in 1997 in Scotland. 1997 Scotland, this was the first case where animal cloning was made successful. Now, in this particular case of animal cloning, although we became successful, do you know how many attempts it took to become successful? because that also speaks about the complexity of this entire process. Can you guess how many attempts it took for us to become successful? Bhima, you are correct. So 100, 200, 500, 10,000. I'm getting all those numbers. So Somo, you are correct. So it just a trivia. If you ever go to KBC, you maybe ask these kind of questions. It took 277 attempts. It took 277 attempts to become successful. How many attempts do you get to become successful in this examination? So anyway, I'll not ask controversial questions. So 277 attempts. So that tells you about the complexity of this entire process. That it's not an easy process altogether, but we have gained success. And ever since, after the Dolly the sheep being the first ever example, we have seen that there have been many such cases in the world and also in India. If we look at the cases of animal cloning in India, the first example of animal cloning in India was of Samrupa, and this was back in 2009. In 2009, this was world's first cloned buffalo. Unfortunately, did not live for very long. Only just after 5 days it was born, it succumbed to lung infections and it died. Then the second case was Garima, and this was world's second cloned buffalo in India, and this was also developed in 2009. This lived longer, lived for almost 2 years, but unfortunately died of heart failure in 2011. And then there was the third case in 2016 of CIRB Gaurav. So CIRB, basically Central Institute of Research on Buffaloes. Now, in 2016, CIRB Gaurav was created, and this was also a cloned buffalo. And we see that this is where it was better this time, that it has lived for longer. And apart from that, there have been other cases. Two of the most recent cases have been these two. One, the case that we saw in 2020, this was done in 2020. Seven clones of M29 Murrah bull and cloned bull of Hisar Gaurav was created. So, so that's why this basically was a case in 2020 where these two cases were done. This was of M29 Murrah bull and this was of Hisar Gaurav. Now, apart from that, last year there was a new case of cloning that has been done, and this time it is a cloned cow calf. This time we have done the cloning of a cow, and for a reason, and there was a reason that was given. The name of the cow is Ganga. The name that has been given to the calf of this cow is Ganga. Now, this calf basically is the first case where we see that cloning of cow has been done in India. And now it becomes a very crucial case because the reason why it has been done, we are saying that this is a first step towards improving upon the milk productivity in India, improving the milk production in India, where we want to rely more on indigenous varieties. All right, we want to uh, uh, build upon the indigenous varieties. So this basically is a species of cow called as the Gir cow, which is an indigenous species. Now, until now, you might have seen that there are a lot of hybrid cow varieties which are foreign varieties, and Jersey cows, for example. So these cow varieties, they are foreign varieties, and they are not uh, easily set in India, and they are not. I mean, when it comes to, let's say, the temperatures and the climatic conditions that we have in India, it's not easy for them to survive in these kinds of conditions, the tropical kind of climate where you have higher temperature, etc. So it's not easy for them to survive in that scenario. But for an indigenous variety, it will be easier. So that's why now we want to carry uh, the cloning of the indigenous varieties of cow and see how it goes and how much success do we get out of it, and whether we are successful in thinking of how we want to proceed here. So that's why we see that there have been all these examples of animal cloning in India. Now, coming to the case of animal cloning, let's also try to understand what exactly is the process. How is this process of cloning conducted? Now, the process of cloning starts with something called as somatic cells. Now, these somatic cells basically mean nothing but all the non-reproductive cells in our body. All the non-reproductive cells in the body, they are called as somatic cells. So, reproductive cells. There are two reproductive cells that humans have. One in the males, that are sperm cells, and then one in the females, which are the egg cells. So, apart from the egg and the sperm cells, the other set of cells that we have in the body, they are called as somatic cells. All right, they are called as somatic cells. So now in this case of somatic cells, what we basically are doing is that this cloning is being performed on somatic cells. They are not being performed on reproductive cells. They are being performed on somatic cells. So once we take that somatic cell, what we do next is that in this somatic cell, out of this somatic cell, we'll first take out the nucleus. As you can see that we have isolated the nucleus out of this somatic cell. Now, once we take this nucleus, all right, once we have taken out this nucleus, this is where our process starts. This is where now we have taken this nucleus, we take an egg cell and we remove the nucleus of that egg cell. And this somatic nucleus will be inserted inside this egg cell. So now you have an egg cell with somatic nucleus. And just think of it, why? Why did we isolate the somatic nucleus? What is the importance of nucleus? Can you tell me this? What is the importance of nucleus? Why did we take only the nucleus out of the cell? Just think of a reason. No negative marking for guessing it wrong. Wonderful. A lot of you are giving correct answers. So Yugnesh, you are correct. Ankush, you are correct. Vaishnavi, you are correct. Madhu, you are correct. Vibhu, you are correct. Shreya, you are correct. Jaswant, you are correct. So more or less everyone is giving correct answers. That's great. So why? Because nucleus contains DNA. We discussed yesterday also. So the genetic material that you have, something that has your individuality, something that has all your characters, all your properties, that's what, that's there inside the nucleus. So that's why if you've taken the nucleus out, you in a manner you have taken the entire characteristics out. The individual characteristics have been taken out by taking out the nucleus. And that is also the reason why in the egg cell also we are removing the nucleus. Why removing the nucleus? Because if you have the nucleus of the egg cell, it means that you have the properties of that female cell, meaning that that donor, wherever you are taking the egg cell from, you have the properties of her, because nucleus is still intact. So that's why we have to take away the nucleus. That's why we remove the nucleus and we replace it with the somatic nucleus. And once we have done that, then activation of this cell is done. Activation of the egg cell with somatic nucleus will be done. Activation means that now we are creating multiple copies. All right, we are creating multiple copies. And once we create all these multiple copies, from here on, there are two things that can be done. Either this leads to the birth of a new animal. For example, what we have seen as the cloning cases. In that case, it is called as reproductive cloning, meaning that this egg cell now will be put inside the uterus of a surrogate mother, and then inside the uterus, the embryonic development will start. So we only have skipped the process of fertilization. All right, we've skipped the process of fertilization, and now directly embryonic development would start inside the uterus of the mother. So this will be called as reproductive cloning. On the other hand, the same cells that we have here, the same cells may not be used for the birth of a new animal, but rather we might want to store them and use them for harvesting stem cells. And we'll discuss that later, how stem cells will come into the picture here. We'll discuss that later. So if stem cells, if let's say we want to produce stem cells out of it, then in that case it is called as therapeutic cloning. All right, in this case it will be called as therapeutic cloning. And if not, if we want the birth of a new animal, it is called as reproductive cloning. So that's the basic difference between what is therapeutic cloning versus what is reproductive cloning. So we start with the somatic cell, and in this somatic cell, uh, we are transferring the nucleus into the enucleated cell, and then the egg cell so obtained will be stimulated with the help of electric shock and chemicals to initiate the division. Within a week, a single cell becomes a ball of mass having around 150 unspecialized cells, and this stage of development is called as blastocyst. This stage of development is called as blastocyst, and once blastocyst is developed, this blastocyst is inserted inside the uterus of surrogate mother to complete the process of embryonic development. So this is basically how we look at the process of cloning. So this is reproductive cloning, and if we don't want the birth of a new animal, then they will be storing this uh, these cells, and they can be used for harvesting stem cells. So reproductive cloning, the newly created embryo is placed back into the uterus where it takes time to develop into an individual. And in therapeutic cloning, it will basically be used for harvesting of the stem cells for medical purposes. So this is the basic difference between what is therapeutic cloning versus what is reproductive cloning. So this question has come in your paper. Very simple question. Please mark your answers. Good to see. 86% of you have marked the correct answer, which is C. What is the application of somatic cell nuclear transfer technology? Somatic cell nuclear transfer. This is what we are doing, that we are taking the somatic cell and we are transferring the nucleus of the somatic cell into an egg cell. So that's why the correct answer will be reproductive cloning in animals. Now the problems, the concerns that we have over reproductive cloning, and especially when we talk about human cloning, that we have seen that in animal cloning we have allowed animal cloning to happen, and animal cloning has been happening in various parts of the world including India. Now when it comes to human cloning, this is where we have seen that there is a kind of a ban that the world has put in, that it has not been allowed to use it has not been allowed to use human cloning, only animal cloning is allowed. Now, why have we done that? There are various concerns that we have with regard to human cloning. So, when it comes to human cloning, what do we see as the problem? Right? There are various problems that we think will face. First is that it might undermine society's respect for human life, that we may not have the same respect that we have for human life, which is being born out of natural processes. And once we know that we can produce new life, then in that case we might not see life as we see it right now. Our entire wiring of the brains might start changing when we think about a new life. So that's why we see that there is one problem that we might face. Then it might also affect social institutions such as marriage and family, that people might think that, do I even need to get married? I can create my own clone and live happily. So in that case, why do I need to get married, and why do I need to start a family? So there might be this structural changes that might start coming in the society. We already see that there are so many structural changes that keep coming in the society from time to time, but this will be a change which perhaps will not be for good. So that's why it will start questioning the social institutions, and that's why the respect for going through with all these social institutions such as starting a family and caring for that family, etc., will start vanishing perhaps. Then reproductive cloning can also make a global security concern, that it may so happen that, let's say, an organization or a country starts thinking of a 15-year plan, that they want that, okay, end of 15 years we want a cloned army. So they'll start the process of reproductive cloning today so that they can have a cloned army tomorrow, and they are the ones that will be used in the armies, or they'll be ones used in the battles, etc. So that might destabilize the entire world as well. And then lastly, it also talks about the concept of designer babies, where we are talking about infusing of desirable traits in the clones, and that's why this is called as designer babies, where you are designing the baby, that you are trying to give the baby, or give the unborn child all the properties that you wanted. So you're looking for certain genes, that you want those genes in that child, and that's why you're trying to replicate those genes only in that child. And that's why this concept itself is called as designer babies. So that also is not good for the society, because then everything becomes commercial, that even birth of a child becomes a commercial commodity, which is not good for the society as such. So that's why we have all these concerns that we with regard to the human cloning, and that's why we see that human cloning more or less has been banned in the world. Then, coming to reproductive cloning versus surrogacy, there is one confusion many a times that we see, when where a lot of students are also confused, that do we mean the same thing then when we are talking about these two things. So now, understand reproductive cloning versus surrogacy, that why surrogacy is different from reproductive cloning. In the process itself, we know that in reproductive cloning we are making the use of a somatic cell, right? And we are trying to create an exact replica of the individual from where this somatic cell has come. But surrogacy is different, because surrogacy still is sexual reproduction. It is still fertilization between the male and the female cell, all right, not being done in a natural manner, being done artificially, but it is still fertilization between the male and the female cell. So that's why in that sense it is different, as a process it is different. Similarly, the uniqueness of progeny, in case of cloning, we know that you are creating an exact replica, whereas when it comes to surrogacy, because it is sexual reproduction, there will be uniqueness of progeny, because there is mixing of genes. Genes of mother and genes of father will be mixed, and hence there will be a unique child who will be born out of it. So that's why surrogacy in that sense will bring in uniqueness, whereas in case of cloning, it's nothing but the same exact replica from wherever the cell has been taken. Then the lifespan. Now, this is a very big challenge and a problem. We have seen that clones do not live normal, healthy life. We have to keep them isolated. We have to keep them under medication. We have to ensure that there are ideal conditions under which they are living. So we have to do a lot of that, and still we do not see them leading a long and healthy life. Still they die young. Many of them have died young. Many of them have not lived a normal life that an individual from that species should have. So there are all these cases, whereas in case of surrogacy there is nothing as such. Surrogacy is as close to natural process that it can be. So that's why there is no problem as such with regard to whether the child will survive or not, or what will be the age, or whether the longevity of the child can be ensured or not. These kind of issues do not exist with surrogacy. And then there are ethical issues, especially when it comes to uh, cloning, human cloning. There are ethical and moral issues that are also raised, which are not so much when it comes to in vitro fertilization. So that's why we see that there are all these things that we need to understand, the various aspects that we need to understand, that how cloning is different from surrogacy. Then coming to the next topic for us is stem cells. Now, this is also a topic which is very often in the news, that you'll keep coming across some news article here and there that talks about stem cells. Now, what are stem cells? Just try to understand what stem cells are and why have they become so important for us? Why do we see them having immense potential in medical science? So stem cells in a manner you can say that they are the origin. They are the origin of any cell in the body, or let's say for every cell in the body. Every cell before it became a specialized cell, it was a stem cell. It's just like how a fully grown child is different from an embryo. where in an embryo, you know that the embryo does not have organs, does not have hands, legs, eyes, nose, etc. Right? But it starts developing, and over a course of time, over, let's say, for humans, over 9 months of time, all the organs, cells, tissues, etc. would grow. All these things will grow into various organs of the body, and ultimately it will grow into a child. But before that, it was just an embryo. Similarly, for any cell, before it specializes into, let's say, a brain cell, a heart cell, a muscle cell, a red blood cell, a white blood cell, a skin cell, so before it develops and develops into a certain cell, into a specialized cell, it would have been what? A stem cell. All right, it would have been a stem cell. So that's why, in that sense, what we see is that they are still stem cells until the time, and then they start developing into a specialized cell. So that's why stem cells, in a manner, you can say that they become the origin of every cell. They become the origin of every cell in our body, and not only our body, in plants and animals also we do find stem cells. So these stem cells, what they do is that they start dividing, they create more of stem cells. So cell division will keep on happening in stem cells, and then ultimately they start specializing into the various different types of cells. And that's where they'll take different shapes and sizes depending on the kind of role that particular cell is supposed to have in the body. So this is how we look at the stem cells. Now, overall, when it comes to the stem cells, understand why exactly are they also important. So stem cells, that's why they, just remember that they can become any kind of cell. The importance starts from there, that they can become anything, they can become any kind of cell. So that's why when people tell you that you can become anything that you want, basically they are asking you to become stem cells. Sorry, bad joke. So stem cells, basically they are the raw material for other body cells. So every cell that we have in our body, they basically come, or they originate from stem cells, right? They originate from the stem cells. Now these, that's why they are unspecialized cells which have not yet developed into mature specialized cells. Now, when it comes to the properties of stem cells, there are two important properties that make them so special, that make them so important for us. First is their ability to self renew, their ability of self renewal, and then their ability of specializing, their ability to specialize. Now these two are very, very important abilities, and that becomes the entire, you can say, fulcrum of why they have become so important in medical science, that why they have become so important for us today, why there are so many experiments and tests, etc., happening in the field of stem cells, is because of these two properties, which are very, very crucial. First, their ability to self renew, meaning that before they start to specialize, all right, before they start to specialize, they first create multiple copies of themselves. They create multiple copies, meaning that cell division keeps on happening. One becomes two, two becomes four, four becomes eight, eight becomes 16, 32, 64, and so on. So that cell division keeps on happening while being undifferentiated, meaning that there would be a lot of stem cells available for medical use with us. So that's why this one property becomes very crucial for us, that they go through multiple cycles or numerous cycles of cell division while maintaining the undifferentiated state. This is called as self renewal. Then, under the right conditions in the body or a lab, they can divide to form more cells called the daughter cells. So that is one property. That's why we see that we use this property so that we can create more cells, we can create more stem cells out of it. And then their ability to specialize. The second is their ability to specialize. Now this ability also becomes very crucial, because in this particular scenario, we know that a stem cell can grow into anything, right? The same stem cell can become a blood cell, the same stem cell can become a skin cell, the same stem cell can become a brain cell, the same stem cell can become a heart cell. So the stem cell has an ability to specialize, that it can specialize into different types of cells. Different types of cells that we have in our body, they can specialize into all these different forms. So that also makes them very, very special and useful for us. So they have the capacity to differentiate into specialized cell types. So that's why this becomes a very crucial property for the stem cells. Now, what we also have to learn is that there are different types of stem cells. We understood that there are two properties that these stem cells have. One, that they have an ability of self renewal, and second, they have an ability of specialization. Now, there are three types of stem cells: totipotent, pluripotent, and multipotent. Understand what we mean. What is the meaning of totipotent or pluripotent or multipotent? Totipotent, in a manner, you can say that toti comes from the word total, meaning that they are the ones that can specialize into all cell types. They can specialize into all cell types in a given body, with the addition of extra embryonic or placental cells as well. So that's why they can specialize into all the different types of cells. All right, they can specialize into all the different types of cells, plus the extra embryonic cells and the placental cells also. So basically, they are derived from the embryos. So that's why they are also called as embryonic stem cells. They are also called as embryonic stem cells. Now, embryonic stem cells, because they can act as an, or they are derived from an embryo. Now, they are derived only during the first two cell division cycles, meaning just after fertilization. Just after fertilization, as soon as cell division has begun, only during the first two cycles after cell division, this is exactly where you have the embryonic stem cells. After that, sorry, this is where you have the totipotent cells. After this, the embryonic stem cells are not totipotent anymore. They become pluripotent cells. So they are also embryonic. They are also embryonic in nature, but the difference being, this is up to first two cycles of cell division after fertilization. All right, and embryonic stem cells, they basically, these are embryonic stem cells after the first two cycles, after the first two cycles of cell division after fertilization. So understood the difference between totipotent and pluripotent. That totipotent is formed within the first two cycles of cell division, just after fertilization, and after the first two cycles, the embryonic stem cells, the stem cells that you are deriving from the embryos, will not remain totipotent anymore. They become pluripotent. Now, becoming pluripotent, they still have a wonderful ability to specialize. They can specialize into all the cell types, but they will not specialize into embryonic, that is extra embryonic or placental cells. They do not have the ability to specialize into placental cells or extra embryonic cells. So they still have a very good capacity, but they still do not have as much capacity as the totipotent cells have. So that's the basic difference that comes in between totipotent and pluripotent. And then after that, we have the multipotent cells, which are also called as adult stem cells. So this is basically, you can say, during or after the birth of a child. Adult means not you have to reach 18 years of age or 21 years of age. That's not what we mean by adult. But what we mean by adult here is that now they are born, that the child is born or the animal is born now. So that's where the stem cell that you derive this time, at this time, when it is not an embryo anymore, all right, development has happened, all the tissues and organs have grown, then in that case they are called as multipotent stem cells. Now these multipotent stem cells, they have a limited ability to specialize. All right, so their ability to specialize is more limited than the pluripotent cells, meaning that they cannot just create anything, they cannot just specialize into anything. Now it depends on where this stem cell is getting derived from. If the stem cell is getting derived from near the heart, then it cannot become a brain cell. It can become a heart cell, it can become a muscle cell of the nearby areas, it can maybe become blood cells from the nearby areas, but it cannot become a brain cell. So now its ability to specialize is more limited as compared to the totipotent and the pluripotent cells. Totipotent could just create anything, it could just specialize into anything that you think of. Pluripotent, all the cell types, but not extra embryonic or the placental cells. But then in case of multipotent, they have a limited ability to specialize, and it depends on where you are deriving the stem cell from. So depending on the location from where the stem cells have been derived from, they would basically form only those kind of cells. So that's the basic difference between these three types of stem cells. Now, when we look at the use of stem cell, we understand that there are various different ways in which stem cells can be used. So we have something called as regenerative medicine. So regenerative medicines meaning that we are trying to regenerate something. We are trying to regenerate damaged tissues or organs. So they can be used to repair the muscles that have been destroyed during an heart attack, for example. So similarly, they can also be used to repair vision. So there are ways in which stem cells can be used. So wherever some kind of damage has happened, maybe internal or external reasons, maybe because of a disease or maybe because of an accident, regenerative medicine they can become, meaning that the stem cells can be used. That whatever is damaged, the damaged cells will be replaced with stem cells. All right, the damaged cells will be replaced with stem cells, and hence stem cells become useful in this particular scenario. Then they can be used in drug testing. That especially what we can do basically is that we can create tissues out of these stem cells which mimic our organs, which mimic our tissues, and once they are created, you can do your testing there. All the drugs, all the vaccines, everything can be tested on those stem cells instead of doing a test on an animal or maybe doing a human test. So instead of doing all that, we can use stem cells for that, and we can artificially grow the stem cells and then do the testing on those stem cells. So that's why they can be used in drug testing or even in vaccine testing. Then in disease modeling. Now, what we mean is that stem cells can be used to create models specifically of genetic diseases, where we can understand what the normal growth procedure should have been versus what is going wrong in the cell growth in the person who has the genetic disorder. So how the stem cells are growing into a defective cell, we can catch that, and that is called as disease modeling. So with the help of disease modeling, we can use stem cells. Similarly, we have something called as stem cell therapy. Now, what we mean by stem cell therapy is that stem cell therapy is where we are in one way or the other making the use of stem cells. So leukemia, lymphoma, blood and immune disorders, thalassemia, in all these cases we see that stem cells have become very effective in treatment of these diseases, where traditionally we did not have medicines, traditionally we did not have treatments, procedures or vaccines. Here we see that stem cells now have started becoming very crucial in solving all these issues. Then they can also be useful in basic research, that we can understand the growth and development and how exactly the life and death is, and how exactly the cells grow and divide and they die, how they develop, what are the things. All these things basically become a part of basic research, where stem cells become very crucial. Then they become very crucial also in tissue engineering. Now, tissue engineering means that we are trying to develop tissues and organs for transplant, and that's why they especially can be used in various different ways, where we can grow these stem cells and we can use them to grow tissues and organs and then use it for transplants. So then in tissue engineering it can be used so many a times again in damaged cells and damaged tissues, etc. We see that they can be very effective in being used. And then there are neurodegenerative diseases where also they can be useful. Neurodegenerative diseases, So neurodegenerative diseases such as Alzheimer's or Parkinson's, etc. In these cases also we have seen that stem cells can potentially be useful, although they have not shown very high success rate, but they have shown success. So that's why they can become crucial in treatment of neurodegenerative diseases as well. So that's why they become useful in all these cases, even in case of autoimmune diseases. We see that we have been trying to use stem cells in treatment of autoimmune diseases as well. So there are all these ways in which they can actually become useful. Autoimmune meaning when immune system itself starts attacking the entire, attacking the self body instead of attacking something which is a pathogen, etc. When the immune system starts attacking itself, in that case it is called as autoimmune. So in treatment of autoimmune diseases also stem cells can become useful. Then lastly, there are certain issues with respect to the stem cells. What these issues are, just try to understand this, that there are ethical issues in the use of stem cell, and this basically comes from the use of the embryonic stem cells. Because in case of embryonic stem cells, you are deriving the stem cell from an embryo, and that is practically killing an embryo, right? It's practically killing an embryo and nothing else. So that's why there are ethical issues raised, and a lot of cultures they believe that life begins at inception, conception, instead of thinking of the person being born. So that's why many of the cultures they believe that uh, the use of embryos means that you are killing a life, something that could have potentially developed into a life, you are killing it. So that's why embryonic stem cells, the use of embryonic stem cells have ethical issues. And then the problem with the adult stem cell, because adult stem cells they have a limited ability to specialize. So on one hand you have ethical issues because of which you don't want to use embryonic stem cells, but on the other hand you don't want to use adult stem cells because they are not that useful for you as much as the embryonic stem cells were. So that's why they, because of the fact that they have a limited ability to specialize, this also becomes a challenge. So there is a way out, and the way out is called as induced pluripotent cell. Induced pluripotent stem cells. Now, the meaning of induced pluripotent stem cells is when you are converting, this is basically converting an adult stem cell into an embryonic stem cell. All right. So converting an adult stem cell into an embryonic stem cell, this is called as induced pluripotent stem cell. So now this is in a manner you are trying to bridge the gap between these two problems. That we are taking adult stem cells, but artificially we are converting them into embryonic stem cell. So that solves both of these problems, right? It solves both of these problems that we have with regard to stem cells. Read this question. And I'll launch the poll for you. Good. So 67% of you have marked the correct answer, which is B. Question says, with reference to stem cells frequently in the news, which of the following statements is or are correct? First is, stem cells can be derived from mammals only. First, we have an extreme word, only. But derived from mammals only is also not correct. You can say that in a manner, the higher animals and plants they do have stem cells. And it's not only animal cells, but even plants have stem cells. So, for example, there is something called as meristematic tissues. So, these meristematic tissues, they basically are found in the of the plant tips of the plant, meaning uh, wherever new leaves are coming from, new flowers or new stems are coming from, these are called the tips. So all these tip that you have in any plant, that is where you have their stem cells that are growing into new types of cells. So that's why even plants have these stem cells. So that's why this is wrong. Stem cells can be used for screening new drugs. We discussed that. We can do it. Can be used for medical therapies called as stem cell therapy. So yes, it can be done. So correct answer will be B, 2 and 3 only. So any question if you have from whatever we have discussed until now? Yeah, meristematic tissues and permanent tissues. So meristematic tissues, yes, they have the stem cell. What is drug screening? So drug screening basically is when you are screening or testing. Screening means testing. So when you are testing the drug, then in that case it is called as screening. Are all the genes of a clone baby same as that of the parent because the sex chromosomes are not transferred? So yes, because we are taking a somatic cell. See, what you have to understand is that we are taking the somatic cell, but every cell in the body will have the 23 pairs of chromosomes. It's not that the somatic cells have only 22 pairs. Every cell in the body will have somatic cells. I mean, every cell in the body will have the nucleus, and the nucleus will contain the chromosomes, and that's why all the genes actually will be replicated. Please leave us early because we have a webinar today. I know you have an economy webinar if I'm not wrong today. Are stem cells found in permanent tissues also or only in the meristematic tissues? In the meristematic tissues, because permanent tissues will not have these kind of growths that happen in the meristematic tissues. The clones of the same parent look like twins. Yes, they would. How many classes of science and technology going to be conducted? A total of 11 classes will be conducted. If natural processes already here reproduction, then what is the need of cloning? See, cloning is done, especially let's say when it comes to animal cloning, we are doing it for certain advantages, you can say, or certain ways in which we can draw some advantages out of this natural process. So, one can be nothing but, let's say, only the genetic preservation, that we want genetic preservation of that animal, and hence we are doing this. Similarly, in the conservation of endangered species, also, animal cloning becomes crucial. Then in medical research and in pharmaceutical research also they become crucial. Then we also try to uh, uh, increase the productivity of milk, productivity of meat, etc., can also be done potentially with the help of cloning. Similarly, to grow new organs so that those organs can be used for transplants, even there we see that the process of cloning can be used. So there are a lot of different ways in which we see that animal cloning becomes useful, and that's why we carry out that cloning process. Is surrogacy possible for any mammal? Yeah, for other mammals also surrogacy is possible. Pluripotent cell and other two types of cell not understood. So basically we are saying that there are three types of stem cells. You have the totipotent cells, you have the pluripotent cells and you have the multipotent cells. Totipotent cells are the kinds of stem cells that have the ability to grow into any kind of cell without exception, any kind of cell. So they are called as totipotent cells, and they are the ones derived from the embryos. So that's why they are called as embryonic stem cells. Similarly, in case of pluripotent cells, they are also derived from the embryos only, but after the first two cycles of cell division. So once the cell division starts, that one becomes two, two becomes four. After that, further division, when it happens, you cannot derive totipotent anymore. The stem cells now found are pluripotent. So you cannot get extra placental cells, etc., extra embryonic cells or placental cells, etc., from these cells, and these are called as pluripotent. And then after the birth of a child or during the birth of a child, the kinds of stem cells that you have, they are called as multipotent. So because they do not have unlimited abilities as pluripotent or totipotent had, because now all the organs and tissues, etc., have grown and specialized. So that's when now we see that the stem cells found now will have a limited ability to specialize. Please repeat cloning and surrogacy. So, surrogacy is nothing but sexual reproduction, where the fertilization is happening between the male and the female reproductive cells, and that's why there will be mixing of genes. And the process of reproduction is similar to how the process of reproduction works in general in mammals. But in case of cloning, we know that it's only one cell that you are trying to replicate with the help of somatic cells. In one movie shown that fetus used in making cosmetics. My god, I don't know of such cases in real life, and I hope there is no such case of in real life. Tomorrow will be class. Yes, I forgot to mention. Please remember, all of you. Tomorrow we will have a class at 5 p.m. The same time as we do usually. We'll start at 5 and we'll end by 8:30. So we will have a regular class tomorrow. It means who look like the same, they have same DNA. See, anyway, if you're talking about a certain species, a huge amount of their DNA will be similar. All their genes will be almost similar. There are only certain differences that they'll have. So yes, if they look similar, then in that case they have some extra similarities. How twins form? No, twins formation has nothing to do with the discussions that we are doing. It is just that when the replication of chromosomes are happening, then the replication does not take a normal course, and then it leads to the birth of twins. That means that human cloning is easy almost via stem cell. We aren't doing because of social issues only. No, stem cell replication is different, and cloning as a process is very different. So, cloning as a process is where we have to put back into the uterus. The success rates are very low. We have to try multiple number of times to get a success. So, that's very different in that scenario. But yes, social concerns are a big reason why it is not done. Somatic cells are nuclear transfer, nucleus transfer. It means the same thing. Is stem cell growing continuously over lifetime? Example as skin? No. So stem cells will convert into or specialize into a specific cell, and these cells keep on developing and dying. Right? Every moment there are new cells being born, they develop, they grow, they die. They are then replaced with new cells. So that's something that keeps on happening. Okay, Yashoda is the movie. I've not seen the film, so I'm not sure what they've shown in that film. But interesting and dangerous concept. Are somatic cells and egg cells both of the same individual? No, not necessarily. It can be taken from anyone. Just that egg cell has come from a female. Somatic cell can come from either a male or a female. Whenever I saw birds, I think that they all are same in physical traits, size, color, etc. But in reality, they are different. Yeah, they are very different, and genetically they will be different. There, and genetic variation, you can say, is the cornerstone of any species development. Any species is able to survive longer if they have good genetic variation. If genetic variation starts to decrease, then in that case it leads to the downfall of that species. So that's why a lot of genetic species is always better for the survival of that particular species. And you look at the species that are facing the danger, and they are endangered species or critically endangered species or near threatened species. They are all the species which in some way or the other they do not have a lot of genetic variation. So that's why genetic variation becomes very crucial for the survival of any species. All right, any other question? Mutation versus variation. So see, the basic difference you can say in mutation and variation is that variations many a times uh, can also be generated because of mutations. All right, because of mutations we might see that this kind of variation might start coming in any given uh, organism. So that's why mutations might bring in variations, and mutations will happen in the genes. Mutations meaning that there are certain modifications or changes happening in the genes, and those changes are bringing in a lot of variations. So you can say that variations in a manner are a subset of mutations. They would happen because of mutations. It's my first class. How can I cover up the leftover classes? You can go through the recordings. You can go through the recordings of all the classes that I've conducted until now. All the recordings are available in your app. M29. I don't know what is M29 in this context. Sorry. Hmm. So, coming to the next topic, and this is DNA fingerprinting. Now, when it comes to fingerprint, what is unique about fingerprint? What is unique about fingerprint? The fact that it is unique for everyone, right? So everyone has a different fingerprint, correct? So when it comes to fingerprints, we know that everybody has a unique fingerprint. Now when we say DNA fingerprint, we know that the DNA structure entirely also is different in everyone. That even if you have identical twins, even if you have identical twins of the same gender, even then they may not have completely similar set of DNA. Still, there will be certain variations in their DNA structure. So that's why the term DNA fingerprint. So it is something that refers to the identification of complete or partial set of genetic information of a particular individual. Now, when it comes to DNA fingerprinting, it can be very useful in a lot of different ways. There are different applications for DNA fingerprint. So, for example, if you see this, uh, let's say we have a crime sample like this, it can help in identification of criminals. It can help in identification of criminals in this manner. That suppose that you collected a DNA sample from the crime scene, and there are three suspects. So, can you tell me out of the three suspects which DNA sequence matches with the one that we found at the crime scene? Which is the one that matches? Yeah, subject 2. So as you can see that this matches, this matches, this matches, and this one doesn't match, this one doesn't match here, this doesn't match, this doesn't match. So in this sense we see that there is only suspect 2 where all the DNA sequences they are matching. So it means that suspect 2 might have been present at the crime scene. We cannot still establish that whether suspect 2 had committed the crime or not, but at least we can establish the presence of that criminal. So now that's why we see that in identification of criminals, DNA fingerprint can be useful. Similarly, in uh, establishing paternity, it can be used because there are certain traits which will be transferred from the parent to the child, and if we see similarity in these genes, we can find the relation between the two. Then claim over the dead body. So in claiming the dead body also, many a times unfortunate accidents happen where uh, let's say the body of the person who died is unrecognizable. So in that case also DNA sequences can be used and DNA fingerprint becomes useful in identification in certain cases. Similarly, making effective drugs. If you remember, we discussed about something called as pharmacogenomics. Correct? We discussed about pharmacogenomics. So pharmacogenomics is where we are establishing the relation between the drug and the genetic makeup of an individual. So when we make drugs according to the genetic makeup, or when we try to understand the genetic makeup and accordingly make medicines, then in that case it is something that belongs to pharmacogenomics. Now, in making effective drugs, it can be very, very crucial, because what happens is that drugs can be developed which are more effective for a particular genetic disposition. And this happens many a times. You'll see that certain drugs are more reliable and they are more effective in some part of the world, and the same drugs are not effective in another part of the world. That a drug that has been manufactured in India, tested on Indian population, perhaps is not having the same effect on African population or on European population, and maybe even leading to certain side effects in them. So because we see that many a times, because of these genetic variations, we have to make drugs accordingly. So that's why in making effective drugs, it becomes very crucial for us to also test it on the genetic makeup. Similarly, treatment of genetic vulnerability, that in this case also we see that a lot of genetic vulnerability can be understood, can be identified very early. It can be identified very early, and that's why it becomes very, very crucial in that sense. So overall, any genetic disease or any possibility of genetic disease can be understood way in advance, and maybe we can start taking precautions accordingly. And then in also wildlife management, they also become useful in wildlife management, because we see that this is where they would become crucial in making in conservation and management of endangered species, and any population where we see a sudden drop in population of the species, we can try to find through their genes what is affecting them, what is causing the problem, what is leading to the decrease in the population that we see in them. So there are all these ways in which we see that in wildlife management also this can be effectively used. So that's why there are a lot of ways in which DNA fingerprinting becomes very, very crucial for us. But at the same time, we also see that there are concerns. There are various concerns that have been raised with regard to DNA fingerprinting. So, understand what are these concerns? What are the issues that we are facing? So, first, information about gene pool can lead to a reference for designer babies, and we discussed about this concept uh, in reference to cloning as well. That cloning, when we are using cloning a lot, it can lead to becoming a reference for designer babies, more so with DNA fingerprinting, because now you are starting to look for desirable traits, that you want some desirable trait in your child, and you're looking for the genes which are responsible for those desirable traits. For example, let's say I want my child to become the best mathematician in the world. So then I'll have to find the genes of the best mathematician of the world, and which are the genes which are responsible for the mathematical aptitude that that scientist or mathematician has. So I'll have to take those genes, and then I'll have to insert those genes while the development of my child is happening in the embryo itself. So once I have these genes, once I insert this gene, the child automatically will have these properties, will automatically will have these desirable traits. So this is where there is a concept which is called as germ line editing. Germline editing. Now, germline editing is a concept where we are putting in these desirable qualities uh, without using the somatic cells. So that's why germline editing can become very dangerous in this particular scenario, because germline editing can lead to the concept, and that's why this concept of designer babies becomes very, very dangerous, because we are editing the germ cells. All right, germline editing means that we are editing the germ cells, and germ cells are the sperm cells or egg cells or even the embryo, that we are doing all the development or all the changes in them. So in that case, what happens is that these changes, once they are done in the germ cells, that is in the reproductive cells, it simply means that they will be passed on from generation to generation, that these properties then will be passed on through generations, and it will not remain localized as was the case with the somatic cells. That in cloning, if you remember, we discussed that we use only somatic cells. Why not reproductive cells? Because if you use reproductive cells, if you use the germ cells, then it means that whatever traits are being introduced, all these traits will become permanent. All these changes will become permanent and they will be passed on from generation to generation. So that is wrong, because then what's happening is that you're not only taking a decision for just one single individual, or maybe just not your child, but instead all your future generations. That I have just decided that I want all my future generations to be very good mathematicians, so I've done a germline editing in the child during the birth of the child, or before the conception itself, to ensure that everyone who is born in my entire bloodline is a mathematician. So that's where this is where you can say that this is also wrong, because you are not taking the consent of the child. You have not asked them whether they wanted to become a mathematician or not. Maybe they wanted to become the best lawyers in the world, but unfortunately you have not given that option to them, because you have inserted this artificially, which will make them mathematician without having a choice. So that's why these can be very dangerous. So that's why when we said germline editing, it is nothing but you can say the modification or modifying the genes in germ cells. So germ cells meaning the egg cells or the sperm cells or the embryos, etc. Modifying the genes in the germ cells of an organism, and just remember that this could this uh, modification, this change this change can be passed to future generations. Can be passed to future generations. So that's why germline editing can be dangerous. Now, germline editing also can be good. For example, if you are trying to treat for a genetic disease, then in that case, germline editing is good. But if you are trying to do this, if you are trying to just uh, create a bloodline with certain properties, then that's wrong. So that's why this is one problem. Then genetic privacy of individuals will be violated. That now what happens is that genetic information is, let's say, widely available. That everybody knows what kind of genes are there in my body. So you know that okay, there are certain genes which are, let's say, desirable genes, or maybe good genes, so-called good genes, and there are so-called bad genes also that are there in my body. And every decision may be based on that in the future. What if, for example, let's say health insurance is not being given to you because there are certain genes that they have found in your body, and now they're saying that no, you have these genes, so that means that this kind of disease can develop in the future, so we can't allow you to get our health insurance. Or they can say that because of this, our health insurance policy for you will be far more uh, costly as compared to what it would be for anyone who doesn't have these genes. So that's why this also becomes a big challenge, right? This also becomes a big challenge, that in the future these all things can be done. that you will not be given loans because of the genes, you will not be given insurance because of the genes, or maybe you'll be charged more premium because of certain genes. So there are so many things that can actually happen in these kind of scenarios. So that's why genetic privacy needs to be taken into account. Then the genetic information can also be misused for commercial purposes. They can be misused for commercial purposes. That again, what will happen is that, let's say, maybe pharmaceutical companies or medical institutions, they might start offering you packages. They might start offering you packages, medical packages, based on the genetic analysis that they have done for you, or maybe the genetic information that is already available, or your genetic information that is already available with them. So they might say that, look, you have this gene. If anyone has this gene, then it develops into a cancer, or it develops into this disease or that disease. Now this is where they will, I mean, especially when it comes to health, everyone will want to preserve their health. Nobody wants to be unhealthy by choice, right? So everybody would want that they are healthy, and if they are being asked to do certain things to become healthier, then they would do that. And especially, let's say, if they instill the fear of death in you, that this disease can happen and that disease can happen, and that's why you need to take this medical package or that medical package, you will do that, right? You will do that to ensure that you don't die, because they will instill that fear in you. So unfortunately, all these things can actually be done in this particular scenario. So that's why you have to understand the aspects of it. And science in general can be misused, and not, let's say, used or misused, whatever you want to call it. You look at all these advertisements out there, cosmetic advertisements especially. You'll see that a lot of cosmetic advertisements, they talk about technologies that you have never heard in your life, and that's why you are impressed by that technology and you go and buy that cosmetic product. I keep giving this example in all my classes, the example of charcoal. That today your face wash has charcoal, your soap has charcoal, your body wash has charcoal, your face cream has charcoal, and everything has charcoal. Toothpaste has charcoal, everything has charcoal. Now why charcoal? Probably many of you do not know that why even we need charcoal. Is there something that charcoal is really doing? Can we start eating charcoal to get all those positives? So charcoal is an antioxidant. To say, let's say, one aspect of charcoal is that they have antioxidants. But do we need so much of antioxidants? Do we need so much of antioxidants? There are fruits and vegetables that are natural sources of antioxidants. Eat them, be healthy. You do not need them artificially. Perhaps in many of the cases you do not need them artificially, but there's a market that has been created that antioxidants are good. You also might have heard of antioxidants in some advertisement. If not, I mean, if you don't know what antioxidants are, you might have heard of it in some advertisement. And every product has an antioxidant in some way or the other. So they're selling it. Similarly, in electronics, you see they'll talk about the technologies that you've never heard of, that this technology or that technology, this is what enhances our product, this is what enhances the user experience and all that. So because we don't understand science, science can be used against us. That you do not understand what is being told you, with whatever they are explaining you, whether that is correct or not, and you believe it because it sounds believable. Who guarantees what I'm teaching in the classroom is correct or not? I am just being confident in sorry, but that's not the case. What I'm teaching is perfectly correct. So, but this is something that can happen. There is a whole book that has been written on this aspect of science, the misuse of science. The name of the book is Bad Science. There's a book, a thick book actually. The name is Bad Science, where the author has taken every segment, that how in nutrition how you are being fooled, how in your diet you are being fooled, how the electronics industry is fooling you, and all these industries how they are fooling you in the name of science. It actually has explained. A wonderful book to read, but I don't think it's the time for you. You focus on studies. Then genetic information can be used to create weapons of mass destruction as well, right? It can create weapons of mass destruction. For example, let's say if you talk about India, we know that the peninsular part of India is protected because of waters on three sides. Similarly, you have the Himalayas on the eastern, northeastern parts. Plus, you have the Karakoram, you have the Thar. So, because of that, you can say that in a manner it has been an isolated area. and there has been a lot of genetic mixing that has happened over hundreds of years. So now obviously there are certain genes that will be common across Indians, across different parts of the country, right? So now when it comes to these common genes, what if tomorrow somebody wants to make a weapon of mass destruction against us Indians? Then, in that case, they have to find those genes which are common only across Indians and attack that. Create something, create maybe a virus, create maybe a bacteria that can attack that. And doing that is possible. With COVID-19, you might have heard that there are many reports that claim that COVID-19 virus was made in a lab in China. Now, whether that was correct or not, we will never know. It's China. Transparent information will never come from China. But we at least have heard these stories and these kind of reports that have been claiming that perhaps that virus was made in the lab. Now, doing that today is possible. We are in a scenario where doing all these things are possible. So maybe it's a possibility, and at least we know that creating a mass weapon of mass destruction is actually possible on the basis of genetic information. So that's why we have to understand all these aspects of uh, uh, DNA fingerprinting. Similarly, we discussed about this yesterday also, that we have something called as gene therapy, where we are making the use of genes in one way or the other so that we can solve a problem. We can uh, diagnose a disease and we can treat a disease. So it can be replacing a gene which is responsible for the disease. It can be about inactivating or knocking out a gene that is not functioning properly, or maybe introducing a new gene into the body to help fight a disease. So we discussed that there are different ways in which gene therapy can work, and today we have seen that there are a number of diseases where gene therapy has started becoming effective, and there is a lot of research that keeps on happening in gene therapy that brings in a lot of different ways, a lot of new ways in which we can make the use of gene therapy. Now, there are certain concerns with regards to gene therapy also. That first we see that gene therapy has a short-lived nature. It has a short-lived nature, meaning that gene therapy, um, although introduced, although effective, hasn't worked for very long in certain cases. And now what happens is that when this new gene, suppose that there is a new gene that has been introduced into the body, all right, once a new gene has been introduced into the body, the gene therapy starts working. But we know that the body also keeps on changing so fast that there are new cells being born every second, and they are getting replaced. They are replacing the older genes or the older cells. I mean, for example, by the time I finish just one sentence, almost 10 lakh cells in my body will get replaced. So that's how fast the replacement rate of the cells work. So what happens in this scenario is that the cells keep on replacing the older cells and new cells are formed. And in this entire mechanism, many a times the body forgets about what was introduced. So that's why the gene therapy does not remain effective for very long. And that's why a person who has gone through gene therapy might have to go through gene therapy multiple number of times. So only going through gene therapy once may not be effective enough. So that is one problem. Second problem has been the immune response itself. That many a times our immune system does not recognize a new gene as friendly, because immune system works on a very simple principle, that anything foreign is an enemy, and that becomes our job to ensure that the immune system does not see this as an enemy, does not attack this gene. So many a times it may so happen that the immune system attacks the genes and then does not let the gene therapy be effective. Then apart from that, there are other disorders which are multi-gene disorders, meaning that only one gene is not getting affected, there are multiple genes getting affected. So that's why in case of, let's say, heart disease, high blood pressure, Alzheimer's, arthritis, diabetes, these are genes, or these are diseases where multiple genes are getting affected. And in this scenario also, gene therapy becomes very complex, that you have to replace multiple genes simultaneously. That's a very, very difficult job. And that's why we see that gene therapy does not remain effective in terms of the multi-gene disorders. So that's why there are all these issues that we have. Similarly, right now we still see that we haven't had a lot of success with gene therapy. We still are trying to strive for success. We still are trying to find new ways in which gene therapy might work, and it also remains very costly. It also remains very costly, and we are trying to reduce the cost. The government of India, for example, the health ministry, along with AIMS, has been working on certain projects where they are trying to decrease the cost of these kind of treatments. But ultimately, right now we know that gene therapy remains costly, and that's why it is not affordable for everyone in the country. Only certain people will be able to afford these kind of treatments, otherwise not. So that's why we know that there are all these issues that we right now have with gene therapy. Then there is this concept of three parents baby. Now, this concept of three parents baby is useful when you have some kind of an issue, especially in the mother cell. Now, when we see that in the mother cell, if there are unhealthy mitochondria, if in the mother cell, if there are unhealthy mitochondria, then this process is used. Now, what happens is, understand this, mitochondria. Heard of mitochondria? Can you tell me what is mitochondria? I know there is one word that everyone is going to use. Yes, powerhouse of the cell. Why do we call them as powerhouse of the cell? So uniquely, I ask a lot of questions which are from class 6, 7th, 8th, 9th, 10th. But there is one question where everybody knows the answer. That mitochondria is the powerhouse of the cell. Yeah, they provide energy, correct. So they provide energy, and that's why they are called as powerhouse of the cell. Now, one thing about mitochondria to note is that mitochondria are always transferred from the mother to the child, never from the father to the child. Mother's mitochondria would be transferred to the child. So that's why if mother's mitochondria are defective, if they are diseased, then the same diseased mitochondria will also be transferred to the child. All right. So child will have the mitochondria from the mother. Always remember this. So um, if that's why if the mothers have, if any mother has an unhealthy mitochondria, then the same unhealthy mitochondria will be transferred to the child. To stop this from happening, this is where we use the concept of three parents baby, where we will have a donor. We will have a donor, and the donor will contribute healthy mitochondria. Donor will contribute healthy mitochondria. So what we do is that we'll take the donor cell, we replace or we remove the nucleus from the donor cell and replace it with the nucleus of the original mother. And why nucleus? We discussed this in uh, cloning also, that nucleus contains all the individual information, all the specific information about an individual. So that's why if you want the properties of the mother, if you want all the genes of the mother, then you need what? You need nucleus, right? So you need the nucleus coming from the original mother. And that's why the from the donor egg, we first replace the nucleus, we remove the nucleus, and this will be replaced by the nucleus of the original mother. And then fertilization happens between the egg and the sperm cells. So this is how this is how we understand that this fertilization happens, and now the child who will be born with healthy mitochondria and having the properties of the original mother and the original father only, and not the properties of the donor. So now this three parents baby concept also has been in the news, that for the first time we see that last year a baby born with biological parents DNA and mitochondria from a donor, this basically has happened. So that's why for the first time we have seen that in the commercial domain also this has been done. So that's why this has been in the news, and it's not that for the first time it has come in the news. For the last 7-8 years it has constantly been in the news, because for the first time in 2016 it was allowed in the UK. So then from since then it has been constantly in the news, and now we see that these processes are becoming useful in solving these problems. So in that context, this question also was asked in your paper, In the context of recent advancements in human reproductive technology, pro-nuclear transfer is used for. So, pro-nuclear transfer is another name for the same concept of three parents baby. And where are we using this concept? Now, I'm sure that this is where everyone will give me the correct answer. So please don't disappoint me and give me the correct answer to this question. Only 66% of you have given the correct answer. When I'm saying that this is three parents baby concept, where are we using it? Prevention of mitochondrial disease in the offsprings. This is the answer. Fertilization of egg in vitro by the donor sperm. The donor sperm is not doing any fertilization. Fertilization is happening between the egg cell of the original mother and the father. Genetic modification of sperm. We are not doing any modification in the sperm. Development of stem cells into functional embryos. Nothing to do with stem cells here. So correct answer is prevention of mitochondrial diseases. It's almost 8:15. You also have the webinar, so let me not start with the new topic. We are left with almost half an hour in this chapter. So we'll do that in tomorrow's class. Please remember that tomorrow we will have a class and we'll complete this portion of biotechnology, and we'll start with a new topic. And now all the smaller topics start. So we'll have nanotechnology and robotics as the next two chapters that we'll do in tomorrow's class. So any questions if you have, please let me know. Yes, same time tomorrow, 5 o'clock, as our normal classes happen. We'll have the same class at the same time. Leaderboard. Leaderboard. Yeah, here's the leaderboard. Congratulations to the four of you who have gotten all correct answer. Is there any gene therapy for sickle cell trait? For sickle cell trait, we actually are focusing more on stem cell therapy. Okay, so do we also have many of you also attending the weekend class? Is that also the case? That you are attending weekday and weekend classes? That I just want to know whether there will be a clash for you. Can you let me know if there are people who are attending both? So a couple of you are. Hmm. We did not think of this. Okay, then let me confirm in that case with the team whether it is possible for us to conduct the class in the evening. Yeah, I know recordings are there, but I don't want anyone to be dependent on recordings in the classroom unless there has been an emergency and you could not come to the class. It's always better to attend the live class. Is this mitochondrial treatment important for babies? What will be the problem caused affecting the mother? So basically, just understand that it will not be for the babies, but rather it will be done before the birth of the child itself, because we would have detected this problem in the mother, and we would carry the procedure accordingly, so that a normal procedure where the transfer of the mitochondria would have happened to the child, we do not let that happen. Let me confirm it whether we can conduct a class tomorrow or not, and we'll inform you via emails and messages if possible that whether we have a class or not tomorrow. Because I see that there are some of you who are attending both weekday and weekend classes. Due to fingerprints, so anyone can identify the thieves. So yeah, that can be done. But what happens is that we also talk about now there is a bill also that is pending, and where we are saying that in the criminal cases we'll start using the DNA samples. So that can become a very authoritative way in establishing that somebody was present. If I have any genetic disease, is it always from my mother's side? No, no, no. Genetic disease can come both from mother's side and father's side. Mitochondrial disease, on the other hand, can come only from the mother's side. So, one thing I'm writing here, mitochondria, mitochondria are always inherited in the child only from the mother, not the father. So genes you get from both mother and father. So that's why genetic disease can be inherited from both mother and father. But mitochondrial disease specifically can be inherited only from mother and not from father. It means that finally living is possible with disease mitochondria. It could be possible. See, it's not. It depends on what kind of disease has mitochondrial traits. So that depends on whether or not that will happen or not. So it completely depends on what kind of disease has appeared and what kind of complexity is there. Sperms do not have mitochondria. No, that's not the case. Sperms do have mitochondria, although their arrangement is quite different. I mean, if you know about the mid piece in the cells, they are located in the mid piece of the cell. So they do have, but arrangements are different compared to other normal cells. What is the main function of mitochondria? Mitochondria provides energy. They provide energy to the entire cell. That is their main function. So for everything that the cell needs to do, the energy that the cell or the cell organelles need to function, that energy they get from mitochondria. That's why powerhouse of the cell. Your way of explaining is amazing. Many topics are just remembered by listening. Thank you so much. I have genetic problem of sinus. Can it be cured fully? Until now, we don't really know of that happening, but we see that these are problems that are inherited. Concerns slide. Concerns slide. Concerns. This one about the gene therapy concerns or DNA fingerprint concern. Any cell in the human body can survive completely individually without tissue system. Actually, they can. I mean, uh, because cells basically you can say that they are a living entity in themselves. So that's why you can say that in a manner they are capable of surviving, although they are in a system that they're living in, and they would be cut off from the sources of food and nutrition. What happens in unicellular organisms, for that matter, is that they have a holozoic system of nutrition in which they are able to eat. But unfortunately, for a cell that we have in our body, they can survive but because they can actually not eat, they cannot get nutrition on their own, that's why they wouldn't be able to live for very long. Hmm. So about the class, we'll confirm you via emails and in the classes, because right now I'll just go and confirm with the team whether we should conduct a class or not. Most probably we will not, because I see that there are many of you who will be attending the weekend classes also. So I don't want a clash for those of you who do that. And as some of you mentioned that I should conduct a poll, it's not about the poll. Even if there are 10 people who cannot attend the class, then in that case it wouldn't be wise to conduct that class. So mostly will not, but let me give you an official confirmation through an email. Right. So let's end the class here. So we'll meet back probably on Monday, and we'll continue from here. Small leftover portion, and then we'll move on to our next segment. Thank you.